Thank you so much. Good morning, everybody. And on behalf of everyone at C-Path, it is really my honor to welcome you all to our first time in years in-person annual meeting here live in person in Bethesda. Good to see you all. So for all of you who were able to make it in person in Bethesda, thank you and welcome. And for a lot of our colleagues who are listening in through Zoom, welcome as well. My name is Kavaljit Singh. I'm the Executive Director of International Neonatal Consortium. And for this event, I'm being helped by my wonderful, wonderful team in pediatrics for organizing all of the events for today and tomorrow. We do have a very action-packed agenda today as well as tomorrow. So I'm not going to take a lot more of your time, except say thank you again and welcome to our 2022 annual meeting. I really do hope you find the proceedings of the next 2 days enjoyable and informative. Now, without any further ado, I would like to invite Dr. Dan Jorgensen, the CEO of C-Path, to open the meetings and provide welcome remarks.
Well, good morning. Thanks, everybody. It's a pleasure to be here at the International Neonatal Consortium annual meeting, and really an honor to share the stage with experts in the field of neonatal medicine from academic, regulatory, biopharmaceutical, and patient care areas, along with patient advocacy groups and patient families. My name is Dan Jorgensen. I'm the new CEO at C-Path. I'm also a pediatrician. I have been on the job for approximately 1 month. I'm fortunate to work alongside a talented and dedicated C-Path team, and I share its mission to improve the health of newborns, infants, and children. As I said, I began my career as a pediatrician with subspecialty training in infectious diseases and a deep interest in vaccines and public health. I worked at the Centers for Disease Control and Prevention and other public health organizations before taking my first job in industry nearly 25 years ago, developing childhood vaccines at Pasteur Marius-Cannot. Subsequently, during my 10 years at Pfizer, I developed anti-infectives in both children and adults at a time of emerging antimicrobial resistance. During this time, I maintained an interest in perinatal and neonatal infections, including the prevention of group B strep disease. What we're doing here at this annual meeting is extremely important. Neonates comprise an underserved population globally. Despite adequate resources, the US has some of the highest rates of infant mortality. Dr. Davis will go into more detail defining the problem, the regulatory and academic efforts, the successes thus far, but also the challenges that lie ahead. This consortium, as you know, is developing the tools and defining the regulatory pathway for the development of safe and effective therapies in neonates. At its core, the INC is all about collaboration. Through collaboration, the INC has shared maternal health data on one end of the spectrum, and on the other end, longitudinal data in infants and young children. Moreover, INC is collecting and sharing real-world data while developing biomarkers and endpoints for clinical trials. Later this morning, we'll get a preview of INC's real-world data and analytics platform. We will also discuss neonatal clinical trial design, placebo optimization, and the use of synthetic and historical controls. INC is growing and changing in a way that benefits newborns worldwide, including important new areas such as genomics and gene therapy. We envision a future where disease-modifying therapies are developed efficiently and effectively for neonates and children of all ages, where cure and prevention are realistic goals. This is C-Path's vision and commitment, and together we can make this happen. In short, this is an exciting time for neonatal medicine, thanks in large part to everyone here today. Thank you for all you have done and will continue to do for newborns, infants, and young children. I look forward to our conversations over the next 2 days. Thank you very much, and I'll turn it over to Dr. Klaus Romero, our Chief Science Officer.
Okay, here we are. So a couple of things. I'm very excited that this meeting is happening in person. When Kanwaljit joined in early 2020, and I was given the baton by Lynn, most of you know her, to take over the role of CSO for C-Path, the first thing I told Kanwaljit is, we're going to have a meeting of this nature that you're experiencing today at the end of 2020. And we all know what happened. So I'm really happy to be here. And what you see today is the commitment we have to the commitment you have, because this is all about the patients and their families and making sure that we accelerate medical product development for this population. And I want to point you to what you see in the graph. Right in the middle of that graph is the year 2015. In 2015, that's when Inc. started. And at the time, as you can see in the scale, is difficult to see for a reason. That's on purpose. We had just about 50,000 individual patient records across clinical trials from industry, observational studies from academia, and some registries. So that was the first example of real world data, and it happened to be in a rare disease. That was polycystic kidney disease. But between 2015 and 2021, Inc. didn't have a single data point in neonates. And that was the other thing that I told Kanwaljit when he joined. We're going to change that. And we're going to make sure that Inc. benefits from the ability that we have at C-Path to integrate all these data. And as you can see, fast forward to the purple bar, the giant purple bar that you see in 2022, and that has kept growing over this year. That's neonatal data alone. That has broken the scale of that graph. That's why the early years are so hard to see and calculate how much data we had when Inc. started. Inc. has now become the consortium that has broken every speed record for getting patient-level data from clinical trials from industry, clinical trials from academia, and John Davis has turned into a force of nature, helping us make that happen. And this is a testament to what? This is a testament to you guys. This is a testament to this collaboration. If we move ahead to the real world data, right? Because Inc. has the ability to combine and integrate data from clinical trials and data from electronic health records. This was the first example at C-Path of true integration of electronic health records. Another thing that I discussed with Kanwaljit was we need to turn around this notion that, and by the way, I'm not a pediatrician, and I get reminded of that every day by Kanwaljit and John and now Dan. But luckily, John has expressed that he can make me an honorary pediatrician. So at least I get that. But one of the things that I want to turn around is this notion that you develop drugs and medical products in adults, and then maybe you think about the pediatric populations, and only maybe once in a blue moon, you think about the neonates. I want to turn that around. And this is an example of turning that around, because what you see on this slide, the purple bar, that has turned things around for other real world data sharing examples. The other diseases that you see there, other rare diseases, have taken the learnings from Inc. and have applied them to their own data sharing endeavors. So this is the first example of the neonatal world teaching something to the other pediatric indications. And I think that is also a pretty good legacy to celebrate. And specifically in rare diseases, beyond real world data, that has also grown, and that is also a lesson that people have embraced from what we have done in Inc. And I think that's pretty fantastic. And what this meeting is going to be about transforming data into insights that we can leverage to accelerate medical product development for this population. But it goes beyond just the neonates, because the neonatal stage of life is a pretty important determinant of what happens later with everything that you experience. And with the way medical product development is evolving, intervening in the neonatal span of the life continuum is becoming even more important. And that's going to be the next frontier for the neonates, teaching everybody else how to do medical product development. And with the tremendous help and support that we get from you and that we get, of course, we are extremely thankful to our regulatory colleagues, our industry colleagues, but everybody in this room, you're making this happen. This is you guys. You should be proud and celebrating this. With that, I'll stop and I'll turn it over to my good friend, John Davis.
Great. Thanks very much, Klaus. And that was a great way to set the stage. And again, welcome everyone. It's really nice to have everyone here in public. And it's nice getting to see a lot of my old friends who don't look so much older after a 3-year hiatus. So that's a good sign. Well, we started out in 2015 sitting around saying, how do we help neonates? We realized that in the United States, as we talked to our colleagues at FDA, that about 6% of all it's about 3.8 million now. The birth rate's actually gone up for the first time in many years, require admission to the NICU. And as Dan said, we have the highest rates of maternal mortality, infant mortality, and prematurity of any affluent country in the world. It's significantly higher in Black and brown communities. So there's a real health equity component to this that needs to be addressed. It's expensive. And despite all our research and efforts, we have not made huge gains in survival and outcome over the past 20 years. And most drugs that we use in the NICU, as my colleagues always remind me, are not approved by regulators for their intended use. And the challenges for caring for these babies really is a global problem, even though the rates of prematurity may be lower, as pointed out by my colleagues in Europe and in Asia and Japan especially. It's still a significant problem worldwide, and that's what we really needed to address. So why is it was so difficult? Well, it was hard to get our sponsors interested in working on neonates. They're small people and it's a small population, and it's not a huge income generating population. And there's small numbers, difficulties with study design, difficulties with outcome measures and assessment tools. It's a vulnerable population, so we needed extra safeguards to really protect this group. It's really hard to establish safety and efficacy in these populations because they have so many problems. And we recognize that the really last drug that was approved in the NICU for use in premature infants that really significantly improved outcome was in pulmonary surfactant, and that was in 1991. So much of the national and international efforts in this area were really being driven by legislation and regulators. And our colleagues at the FDA and EMA, Health Canada, PMDA have all played very, very important roles in driving this process forward. And that's the way it should be. It should be governments really helping us achieve our goals. So here's some of the global legislative efforts to accelerate drug development in pediatrics. I don't have time to go through all of it, but you see there's a lot of different pieces of legislation that we've helped pass to help drive this process forward for children. And we recently saw in the American Academy of Pediatric News a historic milestone. 1,000 drugs, biologics, have new pediatric use information and labeling. That is a huge, huge achievement, because we passed the FDA Safety and Innovation Act in 2012. So in 10 years, this really exploded. But of all these drugs, I'm told only about 50 are appropriate for neonates. So there really are significant opportunities for change and improvement, and no one is driving the process like Inc. is. So this was the first slide that we developed back, and we picked these 8 conditions on the side that we were going to really look at and approach. And we really decided that Inc. should be a safe place for everyone to be able to sit and collaborate and to be a place where we could hear from our parents. We could hear from industry, what's important to them. We could hear from our regulatory colleagues about what's critical for them, and also have a variety of nursing organizations, everybody who's involved in the process of caring for neonates. And we have grown. So we sat in a room in 2015 with 3 or 4 of us saying, how do we pull this off? And I'm really, really proud of this slide and looking at all the groups that have engaged. We have 2 new small biotech companies that have joined Inc. Because they really need help with some of the outcome measures and study designs, and they have access to a variety of different groups here that is unlike anything that exists anywhere else in the world. And that's something we're particularly proud of. This is some of the work group projects that we have had in the past and will continue to work on. As you see from the 3rd one down, clinical pharmacology, and our colleague Bob Ward really helped drive that process forward for all of us. And that paper has been instrumental as the FDA has developed guidance in this area. So that's something that we should all be really particularly proud of, because that was a project where all the key stakeholders sat in a room together and came up with an approach that really has made a significant difference. We're talking about improving neonatal outcomes, and you can see from this slide just briefly some of the papers that we've written. We've written somewhere around 30 or 40 individual papers that have made a huge difference, many times collaborating with our regulatory colleagues as authors, which is even more exciting, and it really has made a difference. And I will point out on this slide to the last one from Jennifer Daigle, the culture of research communication. And it's something that we should all be particularly proud of, because our parents have really put a huge amount of effort into this undertaking. And I can tell you, without people like Deb DeSenza and Jen and others, we would not have achieved anywhere close to what we've achieved. And you're going to hear from Deb. She's our coach, she's our conscience, and she drives the process forward in a way as a member of the leadership team that no one else is able to do. So one of the things that we were particularly excited, as Klaus has pointed out, is we were able to get a grant from the FDA and using real world data to generate real world evidence in neonatology. And you can see what we're working on with this, and you're going to hear a lot more about it, so I'm not going to go into it in great detail. But what I can say to you now is we've gone in the 1.5 years from having no clinical data to having over 400,000 patients in our data set, which is an unbelievable achievement, and working towards getting to my personal goal of 1 million patients, where we can really transform this. And the really exciting part, even more, is that now we're being approached by a variety of different organizations to say, we hear you have this database. So the folks at Columbia and the University of Rochester just recently got a $5 million grant from the FDA to look at anesthesia and analgesia safety in infants and young children. And the first thing they did was approach us to say, could we access your database, see who's gotten surgeries, et cetera. We're also very excited about the process of looking at safety and efficacy of off-label products and simulated control groups for clinical trials, where we might be able to use some of these patients in the data set for placebo controls, cut down the number of patients needed in a trial, cut down the time for the trial, the cost of the trial, something that we're all very, very excited about working with our regulatory colleagues to implement. This is what the analytics platform looks like. I'm not going to go into it in detail because you're going to hear a lot more about it, but you can see on the left all the groups that are giving us data. It has been remarkable. Our colleagues in Japan have been particularly helpful with getting us their data, and a variety of countries and companies and others from around the world. So there's really an urgent need to accelerate drug and biologic development for neonates. Most of these efforts are being driven by legislation and regulators. And for me, personally, the most important aspect is our ability to sit at a table with our regulatory colleagues and ask them for help. And in the past, that wasn't always so forthcoming and easy. But now I think we have a much more collaborative relationship. That all started with Susie McCune at FDA. It's worked on to Jerry Baer and others in her office in pediatrics. People like Ralph Bax is here. Ralph, wave to everyone. Just from EMA has been an incredible help. I can't remember how many emails we've sent him over the last few years asking for his help. He's really been the centerpiece, and Agnes Klein at Health Canada and Junko Sato in Japan have all really contributed in very meaningful ways to the success of this organization. And it's something that I'm really proud of. These multi-stakeholder efforts are essential. We call it in the Clinical Translational Science Award program at NCATS, broadly engaged team science. No one can do this on their own anymore. And the days where I could sit down and write a grant and be the person in charge and get it funded, those days are over. And it was really the grant that we got for our real world data. It was leveraging all the expertise in data science at Critical Path, with Klaus taking a lead role there, with myself and Mark Turner and Tom Miller and others, Mike Padula. And we got together and said, we can write a better grant together than we can individually. And we've made huge progress in a relatively short time frame. And we're really embracing new diagnostic technologies, the real world data, genomics, biologics, really precision medicine for neonates. And we really think this is going to drive innovation in this space. And that's been a tremendous area of excitement for us as how we move forward. And as you heard Klaus say, how do we lead these efforts? And how do we provide an example for our adult colleagues how it should be done? And that's the way it should really start. I look forward to having the opportunity to speak with many of you, but we're really excited that you're all here. Lots of people on Zoom. Feel free to add questions in the chat, and Christine's going to help prime us and prompt us. But thanks so much.
Hello. It's so exciting to be here, and after 2.5 years, I have to say, a little strange. Really I just have such tremendous gratitude that we're able to be here, and that all of you are here, and that we have others who are not able to be here joining us on Zoom. And so I was nominated to give this sort of regulatory perspective, and it feels, like John's talk, a bit of a walk down memory lane. And then it sounds like from here on out, we're going forward. And so I'm going to give you the sort of regulators perspective on INC and what we've done so far. Those are my disclaimers, the usual disclaimers. So INC has created a forum for us to communicate and discuss what's important, what's working in neonatal product development, what isn't working, what are the needs of the families, the clinical staff, the investigators, the sponsors, and even what are the needs of the regulatory bodies. And it goes both ways here. So INC has provided us as regulators opportunities to hear from parents about their families' experiences and what outcomes they value most. And it's also helped us to understand the barriers to communication about trials and recruitment, and what are the challenges at the bedside. And it's also afforded us the ability to understand and even anticipate rapid changes in clinical practice and care standards that are important for us to know in our daily work. And then as regulators, what have we brought to the table, I hope? We've brought to the table a discussion, of what perspectives we need to understand and to do our best and to best do our work. We provide a general understanding of how regulators view the challenges in neonatal product development. And we've also brought the ability to help you all understand the whys and the hows of the regulatory process. So it's been a back and forth, and it's been a give and take, which has been the interactions that INC has allowed us to have really been incredibly valuable. INC has also allowed us to collaborate. These are some vintage pictures. INC has provided this multi-stakeholder forum where we can have collaboration in a product agnostic way, and where we can actually talk about the principles and the concepts and what's important beyond specific product development programs. It's been invaluable to connect us as regulators to families, nurses, sponsors, clinical investigators, so that we can build relationships and that we can better understand each other's experiences and perspectives. We sit around a table, or for the past couple of years, we've sat around a Zoom screen, working together to solve problems and move forward, try to move the needle where it's stuck to get beyond some of our sticking points. It doesn't happen fast. I know some would like things to happen faster. But all the viewpoints are critical, and getting everybody's input is so critical to our success as a group. And then everything old, it's iterative. Everything old is new again. We're revisiting the seizure trials, the seizure protocol that was developed with new information in hand. We're continuing to validate the neonatal adverse event severity scale. We're talking about BPD again, and we're looking at it through a real-world data, real-world evidence lens, trying to answer some of the clinical questions that have long kept us, that have long been sticking points about how to characterize the condition and measure benefit in these patients. And so, it's an iterative process that we've been able to embark upon together. And then the work products, and I won't list those. John had a fantastic slide listing some of the work products, but as a regulator, the work products have been incredibly important. Inc. was C-Path's 9th consortium, and I don't know how many there are now, but it's dozens, maybe. How many? 29 consortia. So we were in some ways we were pioneers. And the mission, as it was written, it was launched as a global collaboration formed to forge a predictable regulatory path for evaluation of the safety and effectiveness of therapies for neonates. And to that end, this group has really built and launched tangible and successful work products like the adverse event severity scale. The hemodynamic adaptation group has helped us to understand how best to standardize blood pressure measurement, and the seizure master protocol. And there are so many others that I don't necessarily need to mention, because John has given us this excellent slide. So overall, regulators have benefited and have hopefully added value to INC through our interactions, through our collaboration, and through the work products that have been generated. I'm going to be the most brief probably. So I would just want to end with this proverb that I think is so appropriate to this group. As John was talking about, he can write a grant and he can get a study done and he can get it published. But in order to really move the needle and improve health for neonates, we need to do this together. If you want to go fast, go alone. And if you want to go far, go together, which really, I think, encapsulates the work that we're doing here in INC. And so I thank you for the opportunity.
Thanks, Jerry. And I'm glad to say, I hope we hang on to those pictures, because it's clear proof that none of us have aged since the beginning of INC. So to your comment as well earlier, John, thank you. So good morning, everyone. My name is Tom Miller, and I am very happy to be here with all of you today. I am the industry representative on our leadership team for INC. It's a position I've held for the past 3 years. I've had a chance to meet many, I suspect not all of you, but I hopefully we'll have the chance to do so during the course of the next 2 days. I thought I would provide some perspective on the sponsor's view, the individuals that have to carry the load within companies. I'm responsible for overseeing pediatric therapeutic development at Bayer, And I think INC has made a meaningful and tangible impact on how we think about approaching clinical trials for this vulnerable patient population. And the way that I think about Inc., one person's perspective, is in three different chapters. In the first chapter, when I joined, we were very much focused on building consensus approaches for trials relating to complications of prematurity. And as I think about that first chapter, and when I joined, I think about using and implementing some of the very tools that were created by this group within our clinical trials for regulatory decision-making purposes. As Jerry indicated, drug development is a long game. It takes quite a number of years to get a drug approved, but having sponsors now implement the great work that you do into trials to standardize our relative approaches that we take, I think is a very important first step. John and Jerry highlighted some elements of what I like to refer to as chapter 2, focusing on new ways of thinking and modernizing our approach towards drug development for this vulnerable population, using real world data and derivatively real world evidence in rare pediatric disease populations. So we embarked upon this journey a few years ago by the very gracious contribution from FDA in a grant that we received to focus on standardizing laboratory values as a function of gestational and postnatal age. And with a second objective of trying to do our best to see if we could define clinical pictures within the data set to define endotypes of disease, most specifically focused on bronchopulmonary dysplasia. That's a great first step, and I'm certain that this will provide value for all of us as we think about drug development in this challenging patient population. But I think the question for all of us today, as we turn the page within this chapter, is can we do more? Where else can we find smarter ways to rethink clinical trial design and execution and operationalization to get to label drugs faster for children? So I'm very much looking forward to these conversations. Chapter 3, as John alluded to, at least from my perspective, was a conversation that began at INC last year. And for those of you that were able to attend our virtual meeting last year, you may recall that I had the opportunity to open the conversation with a not so scientific perspective on the lay of the land in the life science ecosystem. And I indicated at the time, after a review of clinicaltrials.gov, that there were several thousand regenerative medicine studies that were underway at that time. At the time, we had 2 gene therapies approved. We now have 3. And my crystal ball forecast was that that number will grow considerably over the course of the coming years. About 15% of those trials allowed enrollment of children, and some allowed for enrollment down to infancy. So we are seeing now a mega trend emerge with respect to how the industry thinks about development of new drugs, looking beyond just signs and symptoms, disease management, towards potentially curative medicines. The topic of whole genome sequencing is a related topic that will dramatically improve our ability to diagnose genetic disease, with quite a bit of advocacy for implementation of these emerging technologies at birth, perhaps. We very much see a desire to intercept disease in life as quickly as possible, and I very much suspect that this community will have a very important voice in this conversation. So much more on this topic to come, but certainly one that I am particularly excited about, and I certainly hope you all are as well. And with that, I have the privilege to pass the mic to Wakako. Thank you.
Good morning, everybody. I'm Wakako Eklund. Combination of Japanese first name with a Swedish last name, and everybody wonders how that happened. But I think I can represent diversity and equity in a way from a nursing perspective. My slide is very simple, and oh, thank goodness it's not, everything is listed. Okay. I'm a neonatal nurse practitioner. I'm sorry, I did something wrong. Do I click on there? Thank you. Somebody is in control. All right. I would like to just interject a few thoughts as a nurse representative. I'm a neonatal nurse practitioner. I'm still at the bedside. I do quite a few other things on the side as a volunteer, such as here at Inc. representing the National Association of Neonatal Nurses, and also COIN. I'm on the board of Council of International Neonatal Nurses. So today I represent nurses from around the world, from education, such as Carol Kenner is a dean, School of Nursing. I'm at the bedside. Jill Beck is a staff nurse at Thomas Jefferson, but she's also about to become a nurse practitioner. We have nurse researcher, Sheila Gephardt. You will hear from her at another session. And clinical nurse specialist who served at Eli Lilly for many years, Mary. Did I miss any other nursing member? We have the most nursing members represented at this meeting than any other time, and I'm very excited. Now, who are nurses? There's no need for you to even look at that slide, actually. These are just my prompts. So I said, nurses can be in education, research, industry, anywhere. A lot of places have nurses, and we serve numerous different types of roles at various places. And what do we do as nurses in NICU, specifically in NICU? Nurses not only carry the heavy burden of meeting the day-to-day care for the very vulnerable babies that are assigned to them, but also they care for the family members and support the families during the most difficult days, especially when somebody approached them with informed consent, with a bunch of jargons and difficult concepts and everything that scare them to death. And nurses are there, usually watching them mull through, chew through, digest the very complex concept. And by that time, usually the research director or the research scientist who presented the informed consent had already walked away. And nurses are the ones that parents often whisper to and say, what did they really say to us? What do they really want out of my baby who can't say anything for himself or herself? So what are expected of nurses? If they are to support the family, then if they make that decision to enroll their babies, nurses are going to have a whole list of protocols that they must follow, who will have to do a lot more than the standard of care. It could be administering an IV medication or oral medication. It could be following at particular intervals by drawing blood specimen and send it to the lab, not just drawing it. You have to send it to the lab to make sure that it was received at the lab. How many times has that happened to you that nurses tell you that, oh, I did it. For some reason, it's not showing up that it's been received. I have that happen to me all the time. And nurses carry a heavy load. So I would like all of us to recognize the part of the role that nurses play when a very important research protocols are spread. Suddenly, nurses say, I was not a part of any of this decision-making. Suddenly, this is given to us, but they never asked us if this is too burdensome for babies. Is this going to go against the nurse's workflow on day-to-day routine? Or is this going to be a heavy burden on even families who sit by the bedside? If you don't ask the nurses, you may never know how each task can be carried out. So what I mean is, when do you consider nurses? When the trials are planned and designed, you need to get us involved early in your game. And I don't have to say that anymore because many of the members here already recognize that, but I think we need to do that more. And some of the trial concept or the medication you are trying to develop or approve or test it, you may think that nurses just don't get it. Parents won't get it. But if you can't inform the nurses to truly understand that with you early, how can we help the parents to fully come on board and get them to be interested in your research? Because it's not your research, it's our research. And it's our future for the babies, whether we're going to have more options or not. As nurses, we really would love to have more options to help babies. As neonatal nurse practitioners, I'm always aware when I write order for medications that this has been written millions of times in the world, but had not been tested specifically for the baby population. There are so many things we nurses can do for all of us for the future of Inc. Thank you so much. And I'm going to pass it to our parent leader, Deb. So thank you.
Good morning. It's lovely to see people in person in one room. I'm Deb DeSenza. I'm a member of the NICU Parent Network. And to start with, I want to say thank you to Wakako, because as I was listening to her speech, all I could think of was nurses are the trust point in the NICU. That's how you're going to get those patients recruited. So she's very correct. If you don't have the nurses involved from beginning to end, I strongly suggest it. One of the things that, as the NICU Parent Network leader, Kira Sorrells has always said, is that we cannot alone do everything. We have to work together. Together we can make great progress. This is so true. And I'm honored, and I want to include Betsy Pilon and Katie. I'd like you to stand up. They are both members of the NICU Parent Network. You're going to be seeing thank you. We're going to play some videos because not everybody could be here, but we wanted to include as many voices as possible. So I'll speak at the end. Jennifer Deagle is first.
Hi, I'm Jennifer Deagle, and I'm here to thank you all for being at the INC annual meeting today. My daughter was born at 23 weeks gestation back in 2012, weighing just 575 grams. She was 11 3/4 inches long. And she came after having 3 healthy boys, my older son being almost 10 pounds. You can imagine how shocked we were, not to mention that my pregnancy with my daughter was quite complicated. I had a very advanced case of placenta prurita, a lot of trauma and difficult times during the pregnancy, although it was short. And after my daughter was born, she then spent 121 days in the NICU in New York. Her 121 days were full of stress and fear for us. But for her, I could imagine that she endured more medical interventions and trauma than most healthy adults will ever be exposed to in their entire lifetime. And she did not come out unscathed. So besides having some typical micropreemie complications in the NICU, like a PDA, she was septic a few times, ROP, she also ended up leaving with BPD. Her lungs are very, very weak even to this day. And the reason why it's so important to me that I work with INC is because I would really look forward to a world where babies can leave the NICU and not have a lot of the complications that they come home with today. If the lungs could be treated better, if there was medications or therapies that could prevent BPD or ROP, neck, anything like that, it would really make a difference in the lives of these babies. We all know that babies are surviving at much earlier gestations, and they go home to their families who must care for them and have to live with these complications for the rest of their lives as well. I am, again, so sad I couldn't be there with you today, but I know you're all doing great work. And again, from the parent perspective, I want to thank INC for always including us. Our thoughts, our feelings, and our lived experiences are a part of everything that you do. And I know the parents that are there today and for the rest of the workshop will be contributing greatly. And we're always very happy to be a part of anything that is being done with INC. Thank you, and I hope that you enjoy the workshop.
So Jennifer Deagle sits on the leadership team with me. And she's been invaluable, though she's not always been able to attend all the meetings. She listens to the recordings and we talk all the time. Jen is a very hard worker and takes a lot of time out to make sure these things happen. So that she could, if she could have been here, she would have, but she has a full load and a half. I just want to thank her for her hard work. Our next parent is Kaori, and I'm probably going to say the last name wrong, Sakurada, and from Japan. Did I say that right? Yay. Here she.
Hi, my name is Kaori, and I'm a mother of a prematurely born infant. In May 2019, my son was born at 30 weeks at an extremely low birth weight. He spent 5 months in the NICU at Keio University Hospital in Tokyo, Japan. This is where I met Dr. Arimitsu. When my son was in the NICU, I spent a lot of time speaking to Dr. Arimitsu about my anxiety and concerns. I tried to gather as much information as possible that could help our journey ahead. Dr. Arimitsu has always been very supportive, and I've come to realize that he's very passionate about helping babies and their families in the NICU. I've also become very interested in supporting parents in similar situations with my experience. Dr. Arimitsu and I founded the Japanese organization of NICU families called JOIN, along with Dr. Satoshi Kusuda, the director of Neonatal Research Network of Japan. JOIN is Japan's first nationwide network for children and their families admitted to the NICU, as well as connecting parent associations all over Japan. It has been such an honor to be the part of the board members of JOIN. We are passionate about working together with other parent associations and stakeholders to contribute to improve our social, psychological, and mental support. I wish to play a major role in supporting these working groups and raise neonatal awareness in our society. I would also like to learn a lot from worldwide working groups and contribute to advance the field of neonatal therapeutic areas in Japan.
We are very excited to have Kaori involved in our group, and we look forward to hearing from her more. Our next video is from Betsy Pilon regarding HIE, something that we parents don't normally hear about on the prematurity side of things as much. Please make sure you include Betsy in your conversations. And while Katie's here, to walk up to them and talk to them about the different issues. But HIE is something that needs a lot more attention.
My name is Betsy Pilon, and I am the proud executive director at Hope for HIE, the premier worldwide organization working to improve the quality of life for neonatal and pediatric acquired hypoxic ischemic encephalopathy.
My journey into motherhood looked nothing like I thought it would. Max stopped moving when I was 37 weeks pregnant, and we went to the hospital and he was delivered by an emergency C-section. He was transferred down to headquartered hospital in Detroit, Michigan for therapeutic hypothermia. We had no idea that a full-term baby could be so sick, let alone experience a type of brain injury like HIE. While we received excellent clinical care, we felt very isolated as Max was one of the biggest and sickest babies in the NICU, and there was no support or educational information at the time about HIE. Knowing all I know now, I am extremely grateful for the neonatal therapeutic development that led to cooling and that Max and thousands of babies worldwide have benefited from over the last couple of decades. The next question I normally get is, how is Max doing now? I'm pleased to say that Max is a bright, funny, amazingly smart 10-year-old boy who I like to say is living his best life every day. He's not without his impacts from HIE, but he is paving his own way.
Addressing therapeutic gap areas like those that exist in HIE only happens when we can come together around the same table and have all lenses matter and be heard. I'm so looking forward to learning more from everyone at this conference, to speaking at the neonatal neurology section, and to looking forward of how we can continue to advance the field of neonatal therapeutics together.
Thank you. And last but not least, we have Katie.
My son was born at 30 weeks, 3 days, at 1,130 grams in 2009. I experienced severe burnout due to a toxic organizational culture in my executive management career. The career stress of my pregnancy impacted my son's development in utero. During my pregnancy, we experienced a flood in our home. I got called for jury duty. H1N1 swine flu. I fractured my hand and had hand reconstructive surgery. And I developed preeclampsia. I was airlifted in critical condition by fixed wing plane through a blizzard at -1 degrees. This defining moment in our 56 days in the NICU led me to identify the need for credible, trustworthy NICU parent leaders. Meanwhile, my care and concern of our healthcare providers led me to learn that healthcare leaders struggle with burnout and toxic cultures, an issue familiar to me. I left my executive management career and took time to raise my son and pursue graduate school. I am currently a PhD candidate starting my dissertation. As a NICU parent leader, my research and stakeholder engagement focus on the compassionate servant leadership in healthcare cultures. Through seminars and workshops, speaking and coaching, I focus on employee engagement, thriving, and well-being in individuals, teams, and organizations. My son is almost age 13. He struggles with his ADHD, anxiety, and sensory processing disorder. He attends a charter arts academy and loves to play video games with friends. He is on the media team at our church, where he helps film church services and events.
Okay. I'm going to take you back in time to 1963. 1963, you might recall, is the same year that JFK's son, Patrick Bouvier Kennedy, died due to respiratory distress syndrome. That was also the same year my mother was pregnant and lost a baby boy. His name was David Aaron Kiganowicz. My mother very rarely spoke about him. There is no grave, there's nothing. My mother mentioned at one point that had I been born a boy, I would have been named David Aaron. I'm Deborah Ann. 1964, my parents adopted twin boys. They were premature. They were in the hospital for a month. They lived. They took them home and did the best that they could, because special education and other types of therapies didn't exist at that time. They were slow to coming. 1967, I was born. Yes, I'm 55. I'm very proud. I was a month early. I lived. I went home. I still feel the ramifications of that early birth today, believe it or not. Though I did not find out that I was officially premature until after my mother had died. And I saw it in a telegram. Back to 1963. Again, she did not talk about David much at all. How did I find out? There was a hospital bracelet in her nightstand as I was cleaning it out after she had passed away. She had had it the whole time. It had her name on it in 1963. You do not forget the angels. For me, I had Becky in 2003, and she came out feisty and pissed off. And she was 30 weeks gestation. We were told she was doing great. I was very worried about her development. I kept saying this as we took her home on medical equipment with a whole team of specialists. And she landed back in the hospital 5 days later due to feeding issues. That was only the beginning. I thought we were going to be the hospital family. Instead, we were the specialist therapeutic family. She ended up getting diagnosed in kindergarten with autism because I said something, ADHD because I said to the pediatrician, we are talking about ADHD. I had been asking for the better part of a decade about Becky's walking, her gait, her running. No one listened to me the entire time. I went to multiple specialists, developmental pediatrician, an orthopedic surgeon. The orthopedic surgeon must have heard me at 11 years old because by 13 and a half, that's when we got the diagnosis of cerebral palsy with spasticity. I was shocked. 6 months before, her pediatrician told me he saw a curvature in her spine. That's a comorbidity of CP. It didn't take much to connect the dots, and I did. I was furious, to say the least. I was disappointed in the system, the follow-up. I had a child who was suddenly finding out she had yet another disability. What do I do? What do I do for her? How do I lift her up? I'd always done that. And we just picked up and kept going. It's really hard to sit there and say to your child in the car after an appointment like that, Becky, I'm sorry. She's horrified by this. She's quiet about it, but she's horrified by it. I truly believe that we are better together. If we work together, we can make a massive difference. I too am doing my part, not just here yelling at you or pushing you, but also I'm working on a project called Preemie Crystal Ball. It's a data portal, and it works on outcomes data to help everyone, not only from birth, but all the way up through the geriatric years, because we know the effects of the NICU, prematurity has an effect on life. I know that myself. We're all accountable, folks. We all have to do our part. I want to take my data and put it into ink. I'm going to do my part. I thank you for all that you're already doing. Keep going. We have a lot of work to do. This is in memory of my older brother, David, my parents, in honor of my brothers, and in honor of Becky. Thank you.
So that was really wonderful. Thank you, Deb. And thank you everybody who talked about the work that INC has done over the past several years, since inception until now, and the work that we're going to continue doing into years into the future. We're just going to take a quick 10-minute break and gather back here again at 9:15, and where the topic of next session is going to be real world data and the things that we're going to do for INC in the next upcoming several years. Thank you. Okay, well, I think it's time we get started. We've had quite a bit of interesting discussion about the things that INC has done in the past since its inception 7 years ago. We wanted to take some time to talk about the things that INC is doing now and will be continuing to do in the future. Basically, where the future lies. And one of our most impactful projects that John and Klaus actually talked about is our real world data integration effort, which has been going on for the past couple of years with very generous support from FDA, our colleagues from FDA. And we wanted to take a couple of hours to discuss real world data, real world evidence, and how RWD, RWE can play a role in neonatal drug development, where you cannot really use other data types such as clinical trials simply because they don't exist. So in that regard, I am honored to have our friend colleague, Dr. John Concato from Office of Medical Policy at FDA. And he is going to be talking about FDA perspectives about real world data and how real world data and evidence can be used for a variety of different purposes for drug development efforts. So Dr. Concato.
Thank you very much and good morning. I'll start by saying that the importance of this meeting and the work that it represents couldn't be any more clear after our first session. And as we pivot to more on the ground efforts, I just want to say it's a pleasure and a privilege to be here this morning to share some perspectives on real world evidence. The views and opinions of my own are not official FDA policy. I do not have any conflicts of interest, and mention of a commercial product is not an actual or implied endorsement. Let's start by anchoring a topic that you've seen. I think John and others might have shown this slide. The 21st Century Cures Act, more of an inflection point than a true start, but it did mandate that FDA establish a program to evaluate the potential use of real-world evidence to support a new indication for a drug already approved or to satisfy post-approval study requirements. Also mandated, we issued a draft framework in December of 2018 and draft guidance for industry, sort of FDA's current thinking, if you will. And I'll have a slide on what was detailed in September, October, November, and December, respectively. Again, this is technical, but we know we have the Prescription Drug User Fee Act 6 just finished, and PDUFA 7 was just reauthorized by Congress and started October 1st. So I'll say a word or two about that as well. First, just to make sure we're grounded here, real-world data are data related to patient health status or the delivery of healthcare routinely collected from a variety of sources. As shown, we tend to think of EHR claims, registries, but there are other sources such as wearables. And then real-world evidence is defined simply as the evidence regarding the potential benefits and risk of a medical product derived from the analysis of real-world data. A point here in the bottom right portion of the slide is that randomized trials can generate real-world evidence in a strict technical sense. Here's where, for the next few slides, I'll try to provide some insight into what FDA is doing to try to help in this space. But the framework that I described and my comments apply to the Center for Drug Evaluation and the Center for Biologics, and by extension, the Oncology Center of Excellence, whereas the Center for Devices and other centers have their own regulations, their own guidance, not part of my talk today. And you don't really need to know this other than interfacing with FDA in any one of these swim lanes, but our program involves internal agency processes, external stakeholder engagement, guidance development, and demonstration projects. And I'll connect these all to why we're here today. First, in terms of internal and external engagement combined, this is our inner workings, but we're really proud of a real-world evidence subcommittee that's comprised of membership across multiple CDER and CBER offices. That committee provides oversight of policy development on real-world evidence, including guidances. It offers resources and leadership to review divisions. External activities include providing feedback on novel approaches. This is outside of a specific drug application, drug submission, and also discussing initiatives presented to the subcommittee for consideration. And then holding public meetings, actually being here today in a way is part of external engagement. And again, pleased to be here representing a large team. But in terms of how to help sponsors and other stakeholders, these are very technical, but you could see here listed row by row different guidances and categories of data consideration, applicability of regulations, et cetera. So in September of 2021, we published guidance on EHR and claims data. In October, on data standards to get that data into FDA. In November, registry data. Next was regulatory considerations. If a study is an observational cohort study, for example, not a clinical trial, our regulations never anticipated that as well written as they were decades ago. So we wanted to give a sense more than do the right thing, short of revising regulations. And then please be patient, but we do have in clearance guidances on design considerations such as externally controlled trials, which can have a huge effect, a huge impact in many spaces, including neonatal care. FDA supported research projects. Here's where we're getting closer to what we're here to talk about today. We think of them in categories, or if you will, buckets of using or improving the quality of real world data, assessing real world evidence type of study designs, Or the third category, we call them tools. I'll save the left-hand side for last and actually work right to left. Tools would be, for example, if one is not doing a randomized controlled trial, we know that confounding is a problem, but there are tools to evaluate or approaches to evaluate confounded effects. It doesn't mean that we're 100% positive of the outcome, but how large would the effect size of a confounder, excuse me, how large a signal a relationship would a confounding variable have to be to say negate an otherwise statistically significant finding. In the middle category, some fundamental work. The top row again, RCT duplicate is a project funded by FDA, where a team at Brigham and Women's Hospital looked at trials that had both been completed as well as trials that were not yet completed, and tried to emulate the design using, in this case, largely claims and sometimes electronic health record data. Sort of a very basic question of, can observational studies get the right answer when you take your time and emulate a trial? So this is, again, these are representative projects. There are actually over 20 that our office supports alone, but working, again, as I said, right to left, the advancing standards and methodologies in neonatology is my shorthand for the award that C-Path was granted, as mentioned, which is a fundamental foundational work in the real world data category, not only improving the use and quality, but fundamentally first order of business, getting the data in the neonatal space, which had been siloed and unavailable really for collaborative purposes. So actually going back, I thought I had one more slide. We have, just to provide a little behind the scenes commentary, this award was part of a UO1 Health and Human Services mechanism, where it was a very competitive, I'm sorry to say that it was so competitive. It's like NIH pay lines. That's something that is warm and fuzzy, but out of 31 applications, 4 were approved, one of them being the neonatal grant. It's a peer review process. We're very proud of our approach, but the importance, the long-term return on investment, this wasn't very jazzy in terms of there's going to be an immediate return on investment. It was more how over time, if we do this right, if we do the fundamental foundational work correctly, this could provide a huge benefit in ways that we don't know because we haven't thought of them yet, the we being euphemistic. And now let me pivot now to talk a little bit about real world data, real world evidence. If you look back, and I'm not a historian, but real world per Medline is a non-specific modifier, and real world data and real world evidence appeared in the medical literature as of the 1970s or earlier, but in various contexts. I don't think I ever had on a medical school exam, like what is real world data or real world evidence? But now, of course, they have formal regulatory definitions. I submit that older epidemiologic terms were just fine. What's changed here is the emergence of big data and in part the enactment of 21st century cures, other regulatory agencies in other parts of the globe has led to advances, but it's also led to sometimes confusing use of different taxonomies for study design. Like, for example, a real world evidence study is not synonymous with an observational study. You really need the specific details to classify study design. Along those same lines, this slide shows the column headings of drug indication, approval, date, and data. This is not limited to neonates nor pediatrics alone, but notice the column of approval date. Actually, what happened to my slides here? I had a second slide that actually started in 2010 and goes through 2015 to make the point. I think I shortened it to stay on time, my bad, as they say. The point is that we were approving what we now call real world evidence before 21st century cures. This slide, however, let me just go back. There continued is there, yeah, but the first slide is gone. This slide shows in the 21st century cures era, I mainly included, again, not because it's specific. Well, Zolgensma in the bottom row is probably a good one to walk through. That drug was approved for patients less than 2 years of age with spinal muscular atrophy and a specific genetic mutation on the basis of data from a single arm trial compared to data in an external control group based on a natural history study. So the natural history study is where the quote unquote real world data came from. But I want to emphasize that we have a single evidentiary bar on behalf of public health. We're not trying to be difficult. We're not trying to be overly conservative. It doesn't do patients and families any good if we approve a drug that doesn't work. So we have to find that balance point to say that the data are fit for purpose, the study design is adequate, and the conduct of the study met regulatory requirements. Oh, here is the slide I was referring to. It's out of order. This was the UO1 award for the 4 out of 31 applications, but I already mentioned this. And you see the bottom row is the Critical Path Institute with the official title. All right, and this is odd. I'm pretty confident I did not submit the slides in this order, but in any case, here is the 2010. Either that or I was having a bad day at the office. Here is the 2010 through 2015 slide appearing almost randomly. And likewise, here's the slide. Today is an April 1st, right? Here is the slide that shows the 4 guidances again, with the left-hand column being the 2 data source guidances, EHR and claims and registries, and on the right-hand side, data standards and regulatory considerations. I just want to mention a couple of other guidances, again, for those in the field. This is a procedural guidance. It's very short, but you all can help us help you. If in the cover letter of submissions, it was clearer what exactly your data sources and your study design are. Like we have a congressional mandate in PDUFA 7 to track and report on real world data and real world evidence. And I must say, it's quite a challenge because it's a mess right now in terms of how things are described. Here's a slide that represents the progress that has been made in the C-Path U01. I just picked off of a press release from C-Path earlier this year. There's been progress since then, but we were pleased as one of the teams supporting this effort to see how much work had been done to transfer electronic health record data. Dr. Davis et al. Have talked about this. This is odd. My slides are missing. I have a whole chunk of them. Okay, I could ad lib, but it's not quite the same. Should I go forward or backward? Please pull them up. Yeah, I know I probably don't have that much time left, but one of the main take-home points is I'm okay. If there's problems getting it, I have a copy on my on the table. I could just look at it and glance and sort of link my comments to.
And Jen, while we wait for the slides to be sorted, can I ask you to elaborate a little bit on the notion of the fact that this is not new? I mean, as you were indicating, but how the 21st Century Cures Act really put an emphasis on this and what you see as the potential future directions for real world data and real world evidence from the agency's perspective.
I would say that the difference is it's another tool or set of tools in the toolbox. And let me be clear here. Clinical trials should not go away. Randomized trials should not go away. And it's also not as easy. There's no free lunch. It's not as easy as saying, well, we can't do a randomized trial, comma, therefore a real world data, real world evidence study in air quotes is going to be good enough. It has to be robust enough to generate useful evidence. Otherwise, we're doing no one any good, as they say. So I think in the big picture, this should be viewed as a win-win for everyone. But we're trying to help in the real world evidence program by bringing methodologic rigor and holding evidentiary standards so that FDA review divisions are equally comfortable with clinical trial data and with real world evidence data, if you will, or real world data, I should say. Okay, thank you for finding the slides. Again, let's see if I can catch up to where I was and point out the yeah, so these now are in order. Thank you. Where I won't repeat them, but we have the 2 slides. Here's what hasn't been covered yet. Jacqueline Corrigan-Garay and I published a few months ago, Where Are We Now Perspective. And the comment was, the real take-home message was more than 5 years after passage of 21st Century Cures, we still have inconsistent and interchangeable uses which don't help. So we addressed 2 common misconceptions and provided a conceptual overview. I'm rushing a bit, but I'll try to cover these first 2 bullet points. The first misconception, and this is just general background for your awareness, but as I've mentioned, real world data and real world evidence as new concepts, that's a misconception. In reality, sources of data and types of study design, they might have evolved a bit, but they haven't fundamentally changed. What has changed is electronic access to detailed clinical data and the data becoming more relevant and reliable. I remember as a fellow and as a junior faculty member doing paper medical record reviews. You just can't do that at scale. It took us 3 years to do 1,000 patients. And now you hit the F10 key. The question is, though, what's the devil's in the details? What's the quality of the data? Misconception number 2, a simple dichotomy of randomized trials versus observational or RWE studies. In reality, clinical trials are defined by assignment of a treatment according to a protocol. But think about it, single-arm trials face challenges similar to those, right, of observational studies in determining whether differences in clinical outcomes when you compare them to an external control group represent actual treatment effects. So it's more of a continuum than an either or. And this figure brings it all home. It's a little bit jargony, but bear with me for 2 minutes. Randomized interventional studies, non-randomized interventional studies, and then non-randomized and non-interventional studies are the way to think about the lay of the land at the highest level. We often don't talk about that, though, in day-to-day operations. We immediately jump down to a traditional randomized trial, say a trial in practice settings, an externally controlled trial, or an observational study. These are not exhaustive, but these are representative study design types. I think one of the main take-home points is that bracket underneath the central portion of the slide. Real-world evidence is generated when you have real-world data, say at least as the outcome of a clinical trial. Otherwise, a trial that uses real-world data to find patients or sites, my goodness, the streptomycin trial in the 1940s, which is recognized as the first trial RCT in the modern era, they had to find patients, they had to find sites. We don't say that was real-world data, real-world evidence. So it seems silly, but again, with congressional reporting or just when you're submitting something to FDA, please keep this in mind as background. What's more important, however, is this slide, and I alluded to it. This is a very high-level approach, but and reviewers have a tough job, but they do a fantastic job in the review divisions, going through every line of a submission. But if you want to think about a review of systems, if I could invoke my clinical training, first is whether the real-world data are fit for use, and that is mainly reliable data and relevant data to the question at hand. Second, whether the study design can generate adequate scientific evidence. And third, whether the study conduct meets regulatory requirements. This has to do with, say, having the FDA having access to patient-level data. And that worked in this particular example. It's not from pediatrics or neonatology, but Prograf tacrolimus had been approved for prophylaxis of organ rejection in patients receiving liver, kidney, and heart transplants in the 90s based on RCT evidence. And the drug was widely used in clinical care for lung transplants. For various reasons, randomized trials were never submitted to FDA for the lung indication, but the sponsor several years ago developed a supplemental new drug application with a non-interventional study design. The study data and design were evaluated according to our standards, and the drug was approved for rejecting organ rejection and death in lung transplant as of last July. Why did this work? Well, using that same three-level approach, the data were from the U.S. Scientific Registry of Transplant Recipients on all lung transplants in the U.S. for the period shown. So generalizability was not an issue for sure, but neither was validity. The data were similar to what would have been collected if a clinical trial had been done with standard analysis files, et cetera. The design and conduct, this was a non-interventional observational treatment arm. There wasn't even a clinical trial arm compared to historical controls, and the analysis plan and the patient-level data were submitted. We determined the non-interventional study with historical controls to be adequate and well-controlled, or highest evidentiary standard. And we will mention, though, that the outcomes of organ rejection and death are virtually certain to occur without therapy, and that dramatic effect of treatment helped us to preclude bias as an explanation of results. If you have a hazard ratio of 1.1 and a lot of sources of bias, that's not as convincing a scenario, just to be very honest here. And also, to be honest, this is a compilation of many failed attempts to use real-world data, real-world evidence. So this is not about one sponsor or one application, but it does represent lessons learned. What can you all think about so that the mistakes aren't made? In the same three categories of data, design, and conduct, let's start at the top. Issues related to data reliability and clinical relevance. What I'll say there is sometimes sponsors do the best they could for a database that they have available, but ask the question, is the database good enough in the first place? Even if you do the best job possible on a relative basis, on an absolute basis, does it have the data you need? Sometimes there's a need for linkage to other data sources. We don't blame clinicians, right? Missing or mistimed data. Clinicians do not have that Q 3-month, 6-month, 9-month, 12-month protocol in front of them to say, oh, someone's going to be looking at my data later. I better do this test today. And certainly last but not least, suitable capture of endpoints. Endpoints can be an Achilles heel. In terms of non-randomized study designs, of course, confounding and residual confounding, an underappreciated problem regarding index date or zero time, immortal time bias for those who work in the field. Don't ignore that. And then use of an inappropriate comparator. And then in the third category, we really need to know, unlike a clinical trial, I guess there can be fraud and abuse, but that prospective conduct provides a lot of protection. If one has a database and one could hit that F10 key 100 times and change covariates, we need to know that you didn't wait to the 100th until you finally got a p-value less than 0.05 and said, we think we like this particular analysis. Now that's at least mischievous, if not fraudulent, but it's a continuum again. So help us help you by showing us, yes, you have to look at the data to get a sense of feasibility, but you also can pre-specify your plan, tell us what covariates you think are important, and work with that. Almost done, although not forwarding here. Here we go, looking forward. So what is needed to make progress? I think we're at a point in the real world evidence era where we need to, again, focus on the basics. Clear and consistent use of RWD, RWE terminology seems silly, but it's important, at least for efficiency's sake. Attention to fundamental principles regarding data study design and analysis cannot be overemphasized. This is about doing good quality scientific work. And I do think gaining more experience, including seeing the conduct of rigorous non-interventional and interventional studies using real-world data, including what the C-Path U01 will generate, is really going to move the field forward and move the needle. And then last but not least, communication and collaboration with regulatory agencies and other stakeholders. Today is a small part of that, but we try to do our best to do that all year. I want to acknowledge the Real-World Evidence Subcommittee, which again is the center of gravity for all this work. Drs. Cargan Karai and El Zarad in Cedar, the Real World Evidence Analytic Team in the Office of Medical Policy, where I'm located, and then too many to name staff in various centers in real world evidence working groups across offices and centers at FDA. And if there are any questions that aren't answered today, please use the email address shown to reach me, and I'll either answer it or direct it to someone who can. Thank you so much.
Great. All right. Good morning, everyone. Mike Padula from Children's Hospital of Philadelphia and University of Pennsylvania. This morning, I'm going to cover some of the ways that real-world evidence may be used to optimize neonatal care, touching on many of the same themes that John just did, but just putting in slightly different context. I should disclose that along with others at my center, we have received some support via subcontract for our participation in the FDA Real World Evidence Neonatal Pilot Project. So today I'm going to touch on the scope of real world data and show some insights from real world neonatal real world examples that many of you are likely familiar with. I'll touch a bit on some of the frameworks that are used for data aggregation, modeling, and analysis, some of which John touched on. Revisit the concept of the data suitability, or what is fit for purpose. Touch a little bit on some of the analytics generating real world evidence, again, complementing what John just covered. And then I'll walk through a use case that Tom Miller and John Davis had provided, and just leveraging some of those analytic techniques, and then cover a little bit on where we are looking forward. So I know this is going to come as a shock to many of you, but there are several different definitions for real world data. But the consensus generally centers around that it doesn't include randomized controlled clinical trials. What is left is, this is the UK evidence framework says data relating to patient health or experience or care delivery collected outside of highly controlled clinical trials. So that leaves a lot else, from detailed electronic health record data, which I'll touch on again in a moment, to public health data from different registries for birth defects, vital statistics on mortality, immunization registries, infection surveillance. There's also things relating to administrative insurance claims, pharmacy data, device registries, patient registries, whether they're disease specific or various quality improvement registries that many of us participate in. There's also an idea of capturing patient-reported outcomes and surveys, which sort of relate to those patient registries, sometimes not available for our patients while they're in our unit, but maybe potentially later down the road. So I was excited to see Deb's mentioning the crystal ball. And then there are also, in the scope is also things relating to medical devices and social media. Another author had described it as data not collected under experimental conditions, but those data that are generated in routine care. So I wanted to dig into just the sort of vastness of neonatal data that exists within an EHR. These data are granular, longitudinal, largely structured, and compared to what we had probably 15 years ago, many of our centers have much more robust systems on, following infants' growth, vital signs, their entire respiratory course throughout our NICU admission, the medications they receive, and as well as the specifics on their dose interval, what other therapies that they're receiving, and all the relevant laboratory parameters that help us interpret their function. Detailed nutrition data, as well as other details on social determinants of health, things relating to the maternal infant dyad. And then within that, there's a whole nother level of detail that we might be able to uncover with natural language processing, looking at notes from various imaging studies, or the notes documenting our care. So this is touched on elsewhere, but there's a lot of ways these data or evidence can be useful. It can allow us to evaluate therapies in alternative cohorts from different time periods, or slightly different criteria. It allows us to tackle some more rare diseases that we otherwise wouldn't be able to look at when you're able to get larger samples. We can fill in a number of evidentiary gaps that haven't been addressed by traditional trials, particularly uses in patients that were excluded from trials due to other comorbidities. It allows our investigators or sponsors to generate evidence for off-label indications, and hopefully will significantly reduce the time and cost of development for some regulatory decisions, and help us make more informed designs for our future randomized trials. So I'm going to touch on just two brief examples of where some real-world data from a singular source has helped sort of put evidence into perspective. I'm sure many, or if not all of you, are familiar with the Tyson paper that showed that vitamin A intramuscular injection 3 times a week for a month significantly decreased the risk of chronic lung disease or death, and with the number needed to treat being about 14 or 15 to prevent a single case. And this evidence was enough to move many of our centers to use this therapy. But oddly, due to a shortage of vitamin A, almost 15 years later, they tried to reproduce the same criteria that they leveraged in that first study, and actually found that the vitamin A administration was unrelated to death or chronic lung disease. Now, this is not shocking to many people, and it's probably not a question of efficacy of a controlled trial versus effectiveness in a in a larger real-world cohort. It may just reflect the time difference, the fact that 15 years later, our nutritional practices in the US at least, these babies aren't as vitamin A deficient, but it does make you think that a therapy that we once hung our hat on, we may need to potentially serially reevaluate. Another way these real-world data can be useful is antenatal corticosteroids. Up through 2017, the guidance was to use them at 24 weeks on and consider it at 23. And then leveraging the Vermont-Oxford database, where they looked at about 30,000 infants from that 22-week to just under 26-week period. They found that antenatal corticosteroids those infants were more likely to survive than those that didn't receive them, but also received support. Now, based on these and other data, ACOG updated their guidelines to say these may be considered. Now, this isn't, these are just examples of where those data were available and it helped inform decisions. I'm not saying this is the same level of evidence by any means that we would seek for other indications, but it is an example of how as we look at these data that are available, how it can broaden our thoughts for these therapies. So this may be covered elsewhere over the next 2 days, but this is the way I sort of think about steps for curating real world data. So there are a lot of steps involved that you have to typically do for almost every single data field or variable that you collect, where you're cleaning it, looking for to make sure you assess for outliers and completeness. You're transforming it, often codifying data, so leveraging reference terminologies such as SNOMED-CT or LOINC or other sources to transform them to common standards. You normalize the data, and it often also would involve mapping to some type of common data model. It might be an OMAP model, which they use in PedsNet. The Sentinel, which is another example of the FDA of sort of large common data models and processes, and there are others as well. There's steps involved in imputation, so where data might be missing, trying to figure out what makes what might fit for in that case, although you have to be sort of transparent about that process. And also, as I mentioned before, the natural language processing. There's the potential to link data and match patient identifiers, as you group these different sources of data. That can be challenging sometimes when you're dealing with de-identified data that have been offset, and can be a challenge as you try and sort of link back to these data or circle back for these data for longer term outcomes. And then there's a sense of validating the data that, does every variable match expectations based on everything we know about it, the metadata constraints, system assumptions, and what the local knowledge of their sources. So after it goes through that process, and you might see these in different orders or slightly different things, but this is the essence of it, real world data should be ready for analysis. So the concept of what we consider real world evidence, someone bring me back. The concept of real world evidence is quite simply, thank you. You take the real world data, you apply study, design, and analysis, and that's what leads you to real world evidence. It's evidence derived through real world data through the applications of research methods, according to one definition. And often this process can be somewhat iterative, where you can learn something about the process, and then you may circle back on the data to generate additional hypotheses. Now, as I've alluded to this, John Kakata kindly covered some of the framework that is used in the FDA, and there are other guidance. These are just 3 examples, but there are many more on how to address the this process of generating real world evidence. Obviously, the FDA is concerned with evidence for safety and effectiveness, and has provided guidance on alternative trial designs, data standards, and things that support regulatory decision-making. The Duke Margolis Center for Health Policy works very closely with the FDA, as I understand it, and they have a number of similar themes, and also, focus on things relating to fitness for use and the role of reliability and ways to improve those data. And in the UK, there's the National Institute for Health and Care Excellence, who also published their real world evidence framework, where they have identified when real world data can be used to reduce uncertainties and improve guidance. and are best practices for conducting and reporting and including the quality and transparency of evidence. Also relating to this, we had the idea of what makes something fit for purpose. And we've touched on this before, but you have an idea of data relevancy and data quality. Relevancy, may relate to sort of the question, the exposure, outcome, covariates, and whether you have enough, longitudinality to follow them through, as well as the quality of the source data. And how is that governed, as well as is it clear what the methods are to process those data? Sorry. And as I was alluding to earlier, a lot of these concepts relate to one another, and sometimes they have slightly different steps in the process or what have you in the different networks. But they're all centered around confirming the quality in a large sense. And this relates similarly to what John had mentioned, but whenever you consider what data are fit for purpose, you have to consider what question are you asking. And that's really the key to just more data isn't necessarily better. Do you have the right data, and are they credible? Are they reliable? And if there are ways that that you are trying to impute data or adjust for certain measures that those analytic methods are sort of determined from the outset, and you're transparent about what is involved. And John, I borrowed this from him, but it's odd to use somebody else's work and who's in the room while you're presenting, but here we are. I've actually done it twice. The point I wanted to focus on is actually the idea of in the center, the pragmatic trials, which come up again. I had a different slide, but I like this one better, where it's really, the way I think of it is you have the rigor of a randomized controlled trial, but if you want to expand to understand, well, in a not so rigorous cohort with these other criteria, how can I understand how these therapies are actually going to be used? That's where these sort of pragmatic trials can be really helpful to sort of broaden the indications. There are other techniques, and again, this is above my pay grade, but Tom Miller had brought up the topic of Bayesian networks to really find ways to harness these large data sets where you have a lot of confounders. So in short, you can model multiple outcomes and exposures in a single model, and going through this sort of iterative process, you can get a clearer sense, and almost like a learning health system approach, to understand how different exposures and therapies are related, and where there might be evidence to sort of feel more confident. Along those lines, one of the techniques he touches on in this paper are the idea of exploration, where you develop a cohort based on a number of covariates, and you come up with a way to stratify it. If you see that Kaplan-Meier curve on the right, in the different subcohorts, and the stratification shows the sort of distinct risk profiles for a given event of interest, and then you apply it to a new data source to assess its robustness. And if that Kaplan-Meier curve, they sort of, this is my interpretation, if you keep them separate from one another, then you've probably done a better job about keeping those defining those subcohorts. So he also in this paper touches on ways to leverage data from not only real world data, but also existing or historical randomized controlled trials, complemented with newer trials to leverage the fullness of the data. In their paper, they touched on a little bit on one example with pulmonary hypertension, as well as one using ROP, which is in this scenario. So I recommend you take a look at that reference. So, moving forward, it seems that real world data from EHRs, registries, and other sources are really potentially robust for addressing these knowledge gaps in our neonatal care. The techniques for curating and analyze these data to generate real world evidence really require governance and transparency. And I think it's not coincidental that you see so many examples of these different frameworks, because that rigor is really important to ensure that we're making proper conclusions. These data and evidence can complement findings from RCTs and might help with future study design. And I'm still puzzled a bit on mechanisms that we can do to not only ensure the quality, but also figuring out ways how we can link these sort of longer term outcomes back to these sort of de-identified data sets. We'll see how things are moving forward, but I'll love to get people's opinions on that when we have the panel discussion. So with that, I'll conclude. Thank you.
So we are just about to get started with the panel discussion. I see that a lot of people are still outside and getting back in. We are very honored to have a very distinguished set of panelists to talk about perspectives on real world data and real world evidence, specifically as it pertains to neonatal drug development. The moderator of this session is going to be our colleague and collaborator, Tom Miller from Bayer. The panelists are Mike Padula, you've already heard from him, Dr. John Concato, already heard from him. In addition to that, we have Dr. Mark Turner from University of Liverpool, Dr. Ralph Bax from EMA, head of pediatric medicines at EMA, Dr. John Davis, you already heard from him, who is from Tufts University and one of the leaders, directors of INC, Deb Senza, who is one of our parent directors at International Neonatal Consortium. In addition to that, we're honored to have Sheila Gafford, who is a professor at University of Arizona College of Nursing in Tucson. And we're also highly honored to have with us Dr. Hide Nakamura, who is from National Center of Child Health and Development in Japan. So Tom, you are on. Thank you so much again for the panelists, and thank you so much, Tom, for steering this discussion.
Thank you, Kawaljit, and thank you all for agreeing to participate. One of my most favorite parts about planning these sessions, I think is nicely represented on the stage. We truly do have international representation, so from different parts of the ecosystem, and we have all of our key constituencies represented within INC as well. And I think that's really the unique aspect of what contribution we can make or help guide the conversation towards, is making sure that all voices in the ecosystem are appropriately represented. I think the two last speakers, John and Mike, gave a fantastic overview to kind of set the stage for the conversation. The only point that I wanted to maybe emphasize a little bit above and beyond the discussion thus far is examples, more practical examples of how these type of approaches and methodologies could be used. I think first and foremost, the exercise that is the subject of the award that we received, that's been touched upon a few times at INC, is a great example of how to begin the exercise. But I think it's very important for the audience to understand that this is really just the beginning. If we could demonstrate our competency in being able to take seemingly disparate data and combine it for a new useful tool for the entire ecosystem to use, that is a fantastic first step. But this will evergreen over time. We view this as a database that will continue to grow to perhaps exceed John's goal of 1 million patient lives, and will be refreshed and current for everyone in our ecosystem to use to query and ask the important questions that we all have. One of the examples that Mike touched on in my simple way of thinking I like to refer to it as disease endotyping. I think many of the diseases, the complications of prematurity, other rare diseases that may be relevant to some of the regenerative medicines that we'll focus on as we move forward, there are very likely subordinate disease profiles below the common definition that we utilize. I think we all intuitively appreciate this today, but I very much wonder if we can use these large data sources to better endotype patient populations to ensure the patient that we enroll in our studies are a more homogeneous cohort. So if we have a treatment effect with any of our experimental medications, we give it the best chance to potentially manifest. And a little bit later today in the next session, we're going to take a little bit more of a myopic view on one potential area of use of real world data and evidence, and that is alternative study designs and perhaps initial conversations regarding alternative ways to control studies. So let's see how the conversation goes. And so with that, we could go back to the panel slide, just so everybody has a name with a face. Thank you. So maybe to start the conversation with the various constituents that we have today, I'm very curious. So everybody on this panel, I know is quite familiar with this topic because it's been a big area of focus for INC thus far. But what about your broader constituencies? How relevant is this topic? Is it understood? What can we do to educate the communities that we all are responsible in to think about novel approaches beyond traditional study designs as we move forward? So perhaps we could start in linear order. Yeah, you're good.
There's a discussion on how we can utilize real world data, not in neonatology, but in general. And people just think, people just think real world data and electronic health record, then they imagine you can do anything. And, they don't understand the granularity of data, and especially doctors' physical exam information, symptoms and severity related to it. There's not much data in you can extract. And I think maybe I can explain later. Some doctors hospital trying to start a new system to improve that, and that may actually solve the challenge Mike explained in the last slide. But it's nice to really collaborate globally to improve the quality of data.
Thank you. Yep, you're good.
So from a patient perspective, I always feel that the more education you can provide families, the better. It's a partnership between parents and professionals, and it's the same thing with research. So the data can be used to educate in a way that you might not be able to do otherwise just by talking. Partnering with parents is being open and discussing and questioning and all that. It's part of the practice, and I see real world data as part of that. We definitely need to do more education so they understand what it is, because it's just one more thing that they're trying to understand the lingo and all that. That's where the bigger issue is. So
thank you. Mike.
I consider, a lot of my colleagues, that I work with, and I think there are two struggles. One, we have we were to get a sort of tertiary or quaternary kind of center where a lot of rare diseases, and it's sort of been hard to understand what best practice looks like in those centers. So, sources that can really accumulate larger cohorts to better understand it and adjust appropriately, I think would be valued in those diseases. And I think there are also ways that people, have looked to, taking a surrogate marker for, do you have oxygen at, say C-Path for very low birth rate infants. People say, oh, it worked at 28 days or it worked at some interval, but as we looked at more longitudinal data or we use more C-Path, we're not necessarily seeing decreased risks. And so, as they try and tease into what are the reasons why, it might be, I think, look to sources, like real world evidence to better understand this population.
Thank you. Sheila?
Well, I appreciate the question. And in thinking about my constituencies or who I'm representing today, I'm going to put on my nurse hat. There's a lot of expectation in nursing that the data that you enter into the electronic health record would be used for benefit and not that it would just go into this large repository that would then become a garbage can. And honestly, it is an eagerness among nursing for us to learn from our clinical inputs so that we can improve care for babies. So I think that there's a real belief and a hope that people in this room would actually move the needle on that so that the data that we are meticulously collecting would become useful. The other concern I have, though, is from an evidence-based practice perspective in terms of interpreting real-world data, and this could be cross-disciplinary. I think we have some work to do, and it was really helpful to me to hear from John and Mike this morning about these, this is really not new research design. This is a different way of looking at this. And so I think that is an important message that would help in the interpretation of real world data so that it can become useful for clinical care.
Thank you. So Ralph, I know full well that EMA has given this subject quite a bit of thought, but perhaps colleagues would benefit from your insights and state of the art.
Yes, thanks, Tom. Indeed, this is, as you know, a flagship initiative in the European regulatory network. And we are discriminating a little bit between regulators using real-world evidence themselves to support regulatory decision-making, as well as, of course, analyzing and supporting this use of evidence generation as a tool. So what is happening now, we are analyzing, or it has been analyzed, its use for marketing authorizations, but we are also looking at it from a perspective of the pre-authorization space. So here comes into play the pediatric context and pediatric investigation plans, and the post-authorization perspective, of course, where we have been using it most in the safety area. However, I also have to underline, as John has said, it is one of the tools we are developing, and for sure, all stakeholders, including regulators, are, at least in Europe, still on a steep learning curve, and we need to be very mindful that that it doesn't develop in a sort of black box, that everything is transparent and clear, and that assessors, so regulators, understand what it is about. And I think I'm proud to be here because seeing what is happening in the neonatal space, I think we are following a pediatric tradition of pushing for evidence generation. I just remind of extrapolation aspects. Unfortunately, a bit difficult for our space. That is why perhaps neonatology is now spearheading this initiative. Thank you.
Yep, thank you. John, what about your colleagues at Tufts?
So I think this is one of the most exciting projects I've ever done in my career because of the potential applications that we've heard about. I think we recognize that we have to tread carefully. And actually, John has been through this entire process. As I get excited and want to keep moving the needle forward, he brings me properly. We've worked really hard to get this 400,000 plus data set together. And it has been incredibly satisfying as we get these data sets given to us. It's also been the single hardest thing I've ever done in my lifetime. And I think people need to be aware of that. It's a very complicated process. Many organizations don't want to share their data. What? You want us, an organization, I'll give you a perfect example, Kaiser. We've had several discussions with them and they've said, we don't give our data to anyone. They'll take your question and they may work with you to query it if there's money, if there's grants and things of that nature, but it's much harder to do that. Every hospital, company, investigator, everybody you have to negotiate these legal agreements with everyone. And that has been an incredible challenge for us. We'll send them a standard data use agreement from the critical path folks. And if we didn't have the critical path infrastructure to be supporting all these efforts, I would have given up a long time ago, to be perfectly honest. But they schedule the meetings. The investigators love it. They think it's the best thing that they've ever heard. But then they go and send the agreement to the legal department and says, oh, we can't use this. We have our own. And then we go back and forth on the legal agreement. And then once that's signed, we say, okay, we're ready to go. And they say, well, our IT people were really short and we have so many needs and priorities. And so I find I'm calling up the chair of the department to say, you have to push this. So it's personal calls from me to specific investigators at the institutions or at the companies to say, you got to help us because we're stuck in a sort of black hole there. So that's been really complicated. And even then, we sometimes are using grant money to pay the IT departments to actually prepare the data set. And then we de-identify the data sets and it goes to critical path and they may call me and they say, well, you thought it was de-identified, but it wasn't. And partly that's because they took in all the natural fields, the hospitals are taking out the data set, the identifiers. But then in the little free text box that we all have, someone's writing, baby Jones has been on caffeine for 2 weeks. So then we had to start using natural language processing to scan every single electronic health record to make sure that even in those little boxes, the identifiers have been removed. So that's just a small piece of what we've been going through to be able to get those data. Despite that, we've had enormous success, but I'd love to see the process standardized in some way. That every country that we reach out to, every organization is able to use the same real approach. And I think we could use help from our regulatory colleagues or others and governments to say, we have to do this freely and we have to really contribute. And the NIH, for instance, in the US has been great about saying, if we fund you, you have to put the data, make it available publicly, but then the data may not show up for 2 or 3 years after the study is over. So we're waiting, saying, when's it coming out? Well, we're working on it. And it really should be available very quickly and people should be able to use it. I think I'll stop there.
Thanks, John. Sounds like the idea maybe for a future working group. So we'll take a note. John.
Yes, I think in part I answered the question with some of my slides, but I'll add to it. I'm very encouraged that the class is at least half full by the concordance of the responses I've heard. Given my advanced age, I'll just mention what Ralph and John just mentioned. I think you said a learning curve, Ralph, and you mentioned a specific example, John. What I'm encouraged by is how the lessons learned are feeding back to the sponsors. Like even if the first result was negative and their idea failed, to be blunt, they take that back and they're better off the next time. So I think it doesn't happen automatically, but little by little, there are lessons learned if we, again, pay attention to rigorous methods. I also wanted to point out, I didn't have time in my slide presentation, but this panel emphasizes that the international collaborative aspect. That, for example, we have a real world evidence cluster with EMA and Health Canada. We also have international. I gave a talk to at a meeting based in Japan last month. I think it's too soon to talk about harmonization across the board because we don't know enough yet. But I think we can talk about harmonizing terminology at least, and then convergence on approaches, and that can blossom as we go forward. Like Michael, you mentioned about how there are different frameworks. When you look beneath the surface, I think there's more consistency, more similarities and differences. Sometimes terminology, like we say reliability, we're not afraid of the word quality, but if one Googles data quality, quote unquote, you could find 4, 5, 6, 8, legged, 8-item definition. So it's all in the devil's in the details, but I think we're all rowing in the same direction. We're all trying to get at what is fit for purpose data and adequate evidence. And again, as has been mentioned, it's one tool in the toolbox. So we have to be wise about when we use it. I sometimes see sponsors that are so eager to use real world data, real world evidence, and it's not the right time or the place. So it's not about saying, hey, look how great this real world data, real world evidence is. It's more, look how good this study was and how meaningful it is and how helpful it is to improve public health. Thank you.
Thank you. And last but not least, Professor.
Thank you very much. It's great to see everybody. So in Europe, the European Foundation for the Care of the Newborn Infant has set out standards for how kept babies should be looked after. That needs real world data to evaluate them. So the parents are aligned with using real world data. As we've heard, the staff want to do the best, and we need to use novel interventions. So I think everyone's aligned around the need to have high quality data. It's about giving people the rewards so that they know that their efforts are being useful, and that's feeding back to them. I think there are some other things about education. For me, I battle against the evidence hierarchy that has been brainwashed into my colleagues, that you've got to do several randomized controlled trials before you can draw any conclusions. And I think we need to reframe that discussion around the quality of the evidence and the hierarchy of quality, as John laid out, rather than just saying that there's this triangle and you've got to get to the top of it. And that's a big piece of relearning for many physicians. The principles are the same. It's just if you've got something else, then don't always think about a bad RCT when you can do good real world data. In terms of harmonization, there are some opportunities coming across in Europe. People are working on the European Health Data Space, which is developing standards for how people work together. The European Open Science Cloud is moving into that space where you've got to place data. There's a big emphasis on faiриfication, making data fair, findable and accessible, and so on. And that needs a lot of education. And in our Connect for Children project, we've built up a pediatric user guide in CDISC, bringing together all the different data terms there are in CDISC about pediatrics and neonatology, which needs to link up, of course, with the neonatal services in the NCI Enterprise Vocabulary Service. So there's lots of work going on. It's about raising the awareness of it and teaching people how to use it, and that context of it's the quality, the reliability of the data that's important, not just the preconceptions that were bashed into you at medical or nursing school.
Thank you. Maybe let's switch gears a little bit. There's a phrase that I suspect many of you made note of that was used a few times this morning, and as the literature and the how-to papers continue to amass over time, a common phrase that is very relevant for this emerging field is fit for purpose. I think we also used fit for use, but I think it was the same general idea. So maybe for our two presenters this morning, I'm wondering if you could give us, practical guidance for the audience that's thinking about using these data sets for some inclusion in their study ambitions. What exactly does fit for purpose mean?
To me, it's kind of intuitive in that you really, in this vast data set, is really impressive, but you may have a very particular question that you need to answer, and it's whether or not there are the data to answer your question are complete, and you have enough details in terms of adjusting for confounders and other sort of conflicting factors to know that are the sources of data that I'm drawing from, are they of sufficient quality to really answer the very specific question I have? Because it might be good for some context, but not another. Maybe the patient population is rare, or maybe there are too many other details that just may be missing. So it's really just a matter: is it appropriate? Is this data set or these parts of the data set really just appropriate for the question you have, and do you have confidence in the sort of the data that exists?
John.
Yeah, by coincidence, I'll relate a story where I just came back from one of the first trips post-COVID and gave a talk, and it was a year in review that was done the prior year remotely. And last year, I remember a question saying, what is fit for purpose, fit for use anyway? This year, spontaneously, the same speaker said that he's come to understand it. It's a bit of modesty here, but he was complimenting the FDA for its guidances in that regard. So I view it as sort of peeling the onion. If we say relevance and reliability, that's the highest level comment, the highest level category. But if we unpack the relevance, it's really clinical considerations as sort of, the next level down. And then how much time do we have? I mean, we could talk about the study population. Do we know enough about the prognostic characteristics? If we're not going to randomize, and again, I agree with comments, and there's a wonderful article in Social Sciences and Medicine by Angus Deaton and Nancy Cartwright on how to think about randomized trials. But anyway, back to the main point, if we don't have randomization, do we know enough about the variables that predict the outcome in the first place clinically before we get to an analysis? And then if we do know what drives the outcomes, do we have the data in our data set? That's the relevance part, as well as endpoints. You could have, I know there's a slide that some people show that if you have a wearable, you have continuous data, which is wonderful as I live my life, as opposed to if I come in and think of an XY plot where every 6 months. But if that 6 months measurement is valid and verified, whereas the wearable, my wife and I go hiking, we're like on a 5-mile hike, it's a half a mile difference. That's a 10% error. That might not be good enough in a medical context, et cetera. So I think that's an example of how stakeholders, sponsors have to do the work, or investigators, to say what is relevant in this context. On the reliability side, aka quality side, if you don't mind the synonyms that Michael and I use, that's where we bring it down to, again, I would say our guidance for EHR claims is the first one published. It has the most granular details. We talk off the top of my head about accuracy, completeness, and transparency. Each of those needs to be unpacked, but you get the idea, right? Accuracy, are you measuring what you think you want to measure? Whether it's, the wearable doesn't really apply to neonates in terms of step count, but by parallel, you can imagine whether it's in neonatal space or in the geriatric space. If you're looking at Parkinson tremors, how good is a wearable to measure an outcome? And then the completeness is what I had on my slide as missing or mistimed data. It's not the clinician's fault, but when is it good? When do you have the data you need on a timely basis to put together the clinical trajectory? And then last but not least, transparency or provenance. And Michael, one of your slides was it's very similar to what we have in our guidance about following that path from the raw data source, which again was created for another purpose. We get that. Although registries have an advantage where you could sort of script in a protocol, but as you follow, say, EHR claims data, so many steps along the way for things to go wrong, but so many steps along the way for things to be done right, so that going forward we have higher quality data and therefore more convincing evidence.
Great, thank you. Let's pivot a little bit and go back to where we started, and that is the real world data project that is currently underway with INC. I think we'll have 2 working groups, if not this afternoon. I know at some point during the meeting, focusing on both the lab values and the BPD topics. But I'm curious maybe to get the opinion, maybe not everyone down the road, but of each of the constituents. Any thoughts as to where we should go next? Assuming these initial projects will complete, but we continue to grow this data set, hopefully above and beyond the million infant goal. Where else might we be able to apply our data set, and any initial thoughts for the audience to consider? And maybe, if you don't mind, Mark, I'll start with you.
Okay, well, I think one dimension is other topics, other therapeutic areas, such as the hemodynamic side of it, blood pressure, whatever else we want to measure, retinopathy of prematurity. I think there's issues of tractability there. What data is out there? What can be reliably or repeatedly or whatever collected? And I think that there's some issues around that. Some subjects lend themselves more easily to working with real world data than others. So there's one dimension is moving into different therapeutic areas. The other dimension is moving beyond disease progression models into something else. And certainly in the UK, we're in the middle of exploring how to use registries for research. Someone mentioned that randomized controlled trials and registries aren't real world data, but we are experimenting with registry embedded controlled trials and thinking how that can be used by industry and academia. So there are some hybrids in there. So that might be a sort of moving beyond disease progression models into other ways of using data sources might be a more adventurous approach.
Great, thank you. John, I'm sure you have a thought or two.
Yeah, I think there's great opportunities for us. Certainly, as we've gone through different data sets, I've been incredibly disappointed at what's out there. Things like the Vermont Oxford Network, PedsNet, Children's Hospital Association, all of those groups are financially constrained, so they're only putting in data that's very limited. So there's nothing about drug dosages, time, starting, stopping. So we had to eliminate, like 10 million records right off the bat. And then many of the data sets that did have a little bit more granular data didn't have follow-up data to it. So we were very interested in how these kids turned out. We think that's really important. So we're trying to focus on some of those efforts. But I agree with Mark. I think we're already having people approach us. And so the FDA funded another consortium that I'm also part of out of Columbia and the University of Rochester, looking at anesthesia, analgesia, safety, and efficacy in preterm infants, term infants, young children, et cetera. And so we built into the grant using this data set to look at that, because certainly there's been a lot about anesthesia safety in the operating room. And I've said to them, well, sure, but that's only 3 or 4 hours. How about all the babies in the NICU that are on continuous drips for 2 or 3 weeks after their surgical procedure is done, for instance, or other? And so how do we study that, look at that in more detail? So they're very excited. We can look at the doses of drugs that we use and the different drugs. And there's certain drugs used in the NICU without any evidence of safety and efficacy. So we're worried about opioids. So I think there's been other drugs, dextromethorphan, that people are interested in using. And the use of that drug in the NICU has gone up tenfold the last 5 years. Yet there's no evidence that it's actually safe, effective. So, I think there's lots of opportunities. I think there's opportunities to look at best practices. Well, I'm very excited about that. For instance, we've talked a lot to our Japanese colleagues, and they have very, very low rates of necrotizing enterocolitis. And so when I ask Hide or others, or Satoshi, why that's the case, well, they think it's because we have a lot of breastfeeding and diets high in fish and things of that nature, but no one really knows.
Yeah.
And yet they have other complications higher than we have. So why is that?
Yep.
We've all talked about Columbia having University in New York having very low rates of BD. We've all tried to adopt their practices. Same time, they have very high rates of RO. So why is that? If you've done all the right things to prevent lung disease, why are the eyes worse? Not? So I think there's so many questions, and I think there'll be opportunities. We've had calls from industry saying, well, we want to look at different surfactant administration practices and giving it, because people think Lisa, Insure, all these different approaches, yet no one knows whether one actually works better or not. They're all little small trials to do this. Could we look at thousands of patients getting different techniques? Yep. So I think the opportunities are endless if we can keep the process going. And I think that's something that Klaus Walji and I have talked a lot about is, this has been great to have this grant started, but we don't want to say, okay, fine, we'll shut it down. Because now the queries are coming in. So how do we address those queries? How do we prioritize? How do we decide which ones? And continuing to add data as you see.
Yep. And maybe let's stay on the Japan topic, because I think it's interesting because it's a very unique and homogeneous population. Even on with where we're starting with laboratory values, is it a worthy question to ask if normative laboratory values as a function of gestational age and postnatal age may be different in some respects, depending upon where you might be in the world?
Yeah, that's a possibility too. I guess my question is how good the data from Japan in terms of the values and so on. Maybe you can say that in front of other people, but Well, one of the reasons why the data is good from Japan is there are smaller NICUs which are not included in the database, and we don't know about the quality of smaller NICUs, but generally Japanese NICUs are doing fairly good job in terms of medical care. And again, we don't have evidence. I can show you why. And that's why it's important to compare all these data. But the granularity of data is different between database to database. And well, maybe it's not related to what you asked me, but my question is, well, now we gathered all the data and analyze, and there are a lot of data missing, and there are a lot of data which we ideally have in electronic health record. And to gather that data, we'll probably have to change the current way of electronic health record. Do ink go to say that and push the like vendors or whatever, government or whatever, to change the way we gather data? And also, well, we're having the same difficulty in Japan. Medical hospitals think that patient data is theirs. I think that's wrong. All the health record should belong to patients and parents. And we are licensed to use that data or to do the intervention. But, you know, you have to get parent permission to do research. And if we change, you know, and if we all, the whole world agree with that idea, you know, patients carry all the information from different medical institutions, and hospital has the same data, then we don't have to worry about sudden disasters and destroyed one EHR is destroyed, because parents and patients have the same data. And it's about time to discuss that globally, to look at better data structure in EHRs.
Yeah, thank you. And you all are making my job easy, because I had planned on pivoting to Deb next. And wondering, you know, if you could share and provide some of your wisdom, where do you think the story should go next with this evolving data set?
What topics we should be looking at?
Yeah, exactly.
Oh, I mean, it's endless, really. I think it could be very powerful in in drug development specifically. I mean, necrotizing enterocolitis is a lot of what we're seeing as parents as we support families. ROP is another one that's long overdue. And you know, IVH, everything really. But there's also, I know Betsy's still here. I mean, HIE needs some help here. It's not just premature infants. And I think it's really important that we go across different subject matters in the NICU, not just preterm. I know that that's a bigger population, but still, you could go across practice of how the infants are fed, how they go through their feeder grower time frame to go home. And because the feeding issues, I mean, Becky was the same thing. She had to readmit at 5 days discharge. So she ended up back in the hospital because of feeding issues. Those readmits are a big pain on everybody. And we want the kids to go home and stay home. And it causes lengthening of stay. And if we could figure out that magic bullet, so to speak, towards feeding, that would be great. That involves neonatal therapy, a lot of other areas, but drug development wise, I mean, anything, everything. It just, their parents, they're desperate. They're desperate.
Point well taken. And Sheila, from your perspective, you heard John mention a moment ago, one of the things I'm pretty excited about, we're moving beyond the data collection phase to the data insight phase, and queries now beginning to come in. Could you imagine circumstances where your academic nursing colleagues may want to use this type of database for research questions?
Absolutely. Absolutely. I think that in my own story, I was searching for a database when I was working on my dissertation many years ago, and I was, asking all of the questions we're asking today in terms of, the reliability and the relevance and the quality, and is this data set fit for purpose to answer the questions that I have? And I had a hard time finding a good one and went through some of the ones that you mentioned. So yes, there's definitely an eagerness to have access to data like this for querying and asking questions. One thing that I would really be excited about, and we've kind of touched on it, is using these data sources to examine different contextual differences in care. So Deb mentioned it related to feeding and transition to home, but how could we see, who are the top performers? And you gave the example of Colombia with BPD. So, and what's different about them in asking these questions in different ways. But to do that well, I think we need, and perhaps this has already been done, but this is something that I think is essential, is to have some sort of a risk adjustment for different NICU types. Like Vermont Oxford has a good example of that. I know probably the Japanese neonatal network has something like that as well. So working that into the calculation, and then also representativeness of this data is so essential. One of the things I loved about the data set I used for my dissertation was it included, 284 NICUs across the US. And so you may have data sets that are available, but are they representative? Meaning, are we capturing the NICUs where outcomes may not be so much a front and center of their care? Maybe they're just trying to get through the day. So are they possibly not participating actively in some of these quality networks, but they still are caring for these babies who are at such high risk? So I think that's also another thing to consider, especially related to social determinants of health and how we're capturing that and how we're making sure that we're moving the needle on health equity, which I think is directly linked to these contextual differences in care delivery. That's my own angst.
Good. Well, I certainly hope it will be useful for your constituents.
Oh
as
yeah,
well.
that's why I'm here.
Yeah.
That's why I'm here. This is awesome.
Excellent.
I'm really excited about this.
Very glad to hear. Maybe now I'll pivot to a few questions for our colleagues from international health authorities. So, John, you outlined, I think, quite a bit of progress on one of your slides, and Ralph, I'm reasonably up to speed on the guidances that have been issued from the European authorities as well. But at a high level, I'm wondering. One of the challenges that we have as sponsors all the time is that we very seldomly do trials that only focus on one part of the world. We often try to harmonize our studies from a global perspective. So could you give the audience and perhaps maybe most especially the sponsors any food for thought? It seems that in many respects, at least my interpretation, although we are all on a learning curve, we're on the same trajectory in some respects on the learning curve. Are there any areas of maybe where we have a little bit of a difference of opinion depending upon where we are in the world as it relates to use of real world data and or evidence?
Let's start.
Yep.
I would never take the approach of trying to tease out the differences. I'm also not sure, or I'm personally not sure whether we are there yet to tease out differences. I mean, that would be, I just thought during the discussion, actually, we are in a rather good place in going or doing this work together, also from a regulatory perspective. So not, like we are having discussions for the pediatric regulations. Why are there differences and the templates look a little bit different and so on. I believe here we have the opportunity to go this right from the outset together. On the other hand, I'm also envious of the US data set. I mean, I just what you have been describing, I believe we have been trying to interrogate databases also as regulators. And what we see in Europe is that they are mainly based on primary care, so GP data, prescription data, and then even if I go from a broader pediatric perspective, you hardly have any pediatric data in databases. So, and if we now look into the neonatal space, indeed, it would be also from a regulatory perspective interesting to put a query to the INC database. Ideally, we could even, and this is where we are having a potentially more fractured space in Europe with different governments, healthcare systems, to reach something similar. So my approach would be here, if you speak about international development, in how far, and that would be my question, in how far is it actually representative of a bigger development space? Before we go to the world space, let's perhaps look at Europe and US first. And here I'm not so sure. And I think here we have to also in Europe move faster. So perhaps that is what I would see as a difference in not in approach, but in what we have. And my second question is, do you already see any positive outcome of your initiative? So are organizations, are investigators collecting data potentially already in a different way?
Thank you. John, any thoughts?
Yes, I agree that I think in general, the outlook is quite positive. I'm reminded of a meeting, I think it was late June, organized by the EMA and under the auspices of ICMA, International Council on Medicine's regulatory agencies, where a lot of different presentations, over 100 regulatory agencies were present, not all of them presented. But I think conceptually, we're all aligned. I think the differences, again, are sometimes in the nuances of our definitions. And also there's a tension, right? Ultimately, like even let's talk about clinical trials. You could have a review team disagree about the strength of evidence for a small clinical trial with imbalances. So there's a distribution of opinions. And if you start stacking them, what seems like disagreement is really just fundamental scientific, opinion. So I think that's a good problem to have if we're at that point in development. I'm also thinking that what was mentioned, that for decades, randomized trials, to their credit, made such a difference in the latter portion of the 20th century and into the 21st, that we've been trained to think maybe a little bit too monolithically in terms of, no matter what, you have to have like a doubly positive observational study or a doubly negative randomized trial, and then maybe you're on the same playing field, a la grade. If I'm saying that somewhat informally, but those who are aware of the system probably know what I mean. So I think if we recognize that the associations are out there, and there is evolution, like you'll hear later today about different types of trials, but fundamentally the randomized trial is still a randomized trial. We're talking about synthetic patients or external controls or whatnot. So I think we can make great progress again by just trying to understand when we're talking about a drug outcome association, a cause and effect study. Sometimes it's descriptive epidemiology, right? We just want to understand utilization. We now call it real world data. Wonderful. But we were doing those type of studies in the past. The good news is we have the data, which previously was unavailable, to answer some of these basic questions. And I agree, a wonderful use of a registry in one case, in one scenario, is to find patients for a randomized trial if that trial is suitably designed to answer an important question. So I think we just have to connect all of these dots, keep track of these different concepts, even something as simple as validity or generalizability. Someone mentioned, yes, we want a heterogeneous data set. I don't mean to be devil's advocate, but in some cases, getting a narrower answer, I don't mean like ethnically, racially, but if you have a homogeneous population available, especially with real world data, real world evidence, do the analysis, get that answer, and then maybe the next grant, Dr. Davis et al. Could be to expand to generalizability. So I think there's a tension, but it's a healthy tension, an inherent tension between these different competing demands, and we just have to make the best decision on a step-by-step basis.
And Tom, I'll just add to that. One of the unique aspects of this data set is we have so many different sources of data. And so we have electronic health record data, registry data, randomized controlled trials from industry, academic investigator trials, and being able to look at all those data sources, I think is going to be one of the more interesting parts to see and really educate our regulators on what kinds of data can you really rely on. And some of the people who have donated their data have said, you know, our data is so good. We spent so much time cleaning this data and working on it. And then I hear, well, maybe it's not quite as good as they thought. But, you know, if we can then educate different investigators and companies on what is really good data, and you need to get that up front and not wait till afterwards. I find that too many clinical trials, we spend 6 months afterwards, quote, data clean. And that was a real challenge. One of my trials, they didn't realize till the end when we were looking at laboratory values that the Europeans used one set of metrics and we used a different type in the US. And they were getting ready to close the data set and said, oh my goodness, now we need to go back to the beginning and transform every single lab value so we can compare the set. So, you know, I think people don't really appreciate the nuances. And that's to your point, John, that, you know, we may need to focus on one and ask a question and build it out there.
Okay, so maybe one last question before we could open up for any audience questions or anything in the chat as well, as if that's being monitored. And I'll, oh, sure, please.
As parents, this is a question that, can you hear me? I just wondered how involved are you in some of these clinical trials and in trying to help enroll or to improve the participation of the eligible participants? Do you talk to parents? I mean, had there been any possibility where you could do that or be a team of a research team?
I would love to do that. Right now, I'm supporting parents and I'm trying to direct and navigate what they need right now. If someone were to approach me and say, hey, we're trying to recruit for this trial, you know, I'd be happy to say, hey, here's some tips on how to get the parents involved and get them on your side. Because it's all about trust. You all heard me talk about how nurses are a trust point. I will emphasize that again and again and again. They are your friends, they are your allies, and they can get the parents on your side. As far as the clinical trials go, you know, we parents are not always approached. We're given flyers and things like that to put out on social media. And it's not really in depth. It's not as in depth as I think it should be. But yes, that's pretty much how we.
Sheila.
I would just operationally give this input that if you form a parent advisory team with your trial team and have them be locally and embedded in wherever you're actually recruiting, that can be very effective.
But Sheila, help the companies that are running trials in multiple hospitals and areas. How do they do?
So I don't know exactly. I've seen some connections through NICU Parent Network, but they'll tell you to go ahead and create your own team and then to link with the family support teams in the unique hospitals. If you're enrolling in a hospital context, which I would imagine most would in a NICU context. So a lot of hospitals will have parent advisory boards that can be a resource and at least give input on what will work in their unit.
Is it a bit late when you're getting to the individual hospitals? It's best to have parents involved in the design of the studies to make sure they're meeting important endpoints, that they're acceptable at every stage. And I mean, we'd always try and build at least one parent into the trial management group, the project steering committee, all those governance bodies, and have people thinking about the design of the study before it goes into the PIP or before it goes into the protocol, so that it's got a chance of being acceptable.
Yeah, I like that. I would just encourage the input I got from Kira Sorrells from NICU Parent Network was to always do at least two parents. So, and to think about them being support for each other.
And also the fact that you can, we have always discussed in NPN and I've always said, wouldn't it be great to embed one one of our members or somebody into the recruitment to be there as a support for the parent. Not that they understand the study and they have all that wisdom, but they're there as a support, because that's a hard decision to make.
Other question? Oh, please continue.
Dr. Padula reminded us about John Tyson's wonderful paper on vitamin A in 1999. He further showed evidence using real-world data, however, within an evolved NICU that confirms that data. My question, however, is, what is the concordance of a clinical, known clinical trial with the use of a real-world data with the same kinds of conditions? There are so many trials in newborn, CAP trial, TIP trial, ENOS trial. And you could do this, and can you replicate that using real-world data, using the same kinds of conditions? I'm still wondering how close are they? Of course, randomized clinical trials is really not a pragmatic trial. So there may be differences, but I'm just wondering, we keep talking about real world data as though it is the gospel truth, because that's what's happening to thousands of people. But are they really comparable?
Do you want to go first? Since you brought your name up?
Yeah, sure. I'll be ready, Mike.
To me, it's just I wasn't suggesting that it's no longer, like, I didn't mean to suggest that that it doesn't have a role or it still may not be value, or to point out that there may be, some signal that other practices have changed over time, and so we may not get that same benefit that we saw before. And I don't know all of this literature as well as I could, but I think there've been trials with enteral vitamin A, as well as looking at well-resourced versus less resourced in NICUs across the world, and those areas where the nutrition practices might not be as strong, then we might see more benefit. But the take-home message for me is, where our environments that we practice in are not static. There are a lot of variables that are changing over time, and so to be able to sort of check in with these therapies and just sort of see how they play out in different cohorts with real world data, to me is intriguing to sort of better understand sort of how our care is evolving over time, and where there are opportunities still to improve.
I would just add to what Michael said. I think two things. One is that we've known, let me start with, it's easier to mess up an observational study, right, than a clinical trial. There are many more things that have to go right to get a valid answer. So we're not saying that you just wake up in the morning, grab a data set, come up with a design and analysis, again, hit that key and get results. Having said that, we've also known for more than 20 years that well-designed observational studies can replicate randomized trials. And most recently, one of our demonstration projects that I had as one bullet point on one slide, it's in the adult space. It's not even in peds, it's not in neonatal. Now, are there enough trials and enough databases? Maybe in the future, we could look into that. But when trying to emulate a trial, and that's also been known for 20 or 30 years, there's some recent work by Renan Robbins et al. That's getting a lot of attention for good reason as one approach to trial emulation. But the idea is we should think about how a trial was designed. But again, you need the data on the covariates to affect that balance. I will also point out what's underappreciated. Randomized trials on the same topic very often disagree. They're actually more prone to disagreement because you have different patients based on inclusion-exclusion criteria. You have different mode of therapy, or details of dosages of therapy or concomitant therapy. And in some cases, less often, but in some cases, the outcome is not the same. So I think we have to be humble enough to know that a single study of any sort doesn't find capital T truth, with rare exceptions. Actually, the exceptions I'm thinking of weren't randomized trials at all. Banting and Best, when they first lowered glucose in a beagle, didn't need a control group, didn't need randomization. That was deterministic. Very few things in our line of work are quite that easy. But I think that if we take a agnostic view and keep our evidence bar high and understand that randomized trials are the archetype type of study design, but they're not the only study design, we can make progress with using the right tool at the right time and not forcing a screwdriver when you need a wrench or vice versa.
And I'll just add 2 examples of that, Jack. I sat on the FDA committee initially that approved progesterone, the emergency use authorization for prevention of premature labor, and it was done by the NIH, network, and it was a great study. But FDA, in giving that emergency use authorization, required one more study to confirm it. The problem was that it became standard of care in the U.S. and no one was willing to do the trial here, so they moved it to Europe and Asia and other countries, and they couldn't find any benefit to it. So I know there was another panel recently that recommended removing it from the market. The other example is antenatal steroids. I mean, that's as Mike said, that's our gold standard in US and Europe and Asia, et cetera. And yet the Gates Foundation did a trial in Africa and low resource countries and found that it was actually harmful and not beneficial at all. So now they're redoing that study again with some tighter entry criteria. How do you explain that? I mean, that's why it's complicated. And I think having these other data sources on top of randomized controlled trials, exactly what you're suggesting, John, will be really important so that maybe we don't have to go into a big trial to find that it doesn't work, but we can use sources of data to question whether it's working or not.
John, do you agree? We hope that future generations in general and in scientists and medicine in particular, figure out what we did wrong. Our job is to use the best available data, the best available designs. We do make at FDA, since I'm very humbled by the idea, we make point in time decisions based on the available data and evidence. And we just have to understand that that is in the interest of public health. And if we figure out things later because we don't understand it today, that's just good for our children and grandchildren.
Yep, I think all very fair points. Please. Oh, sorry, Mary, just one last point. So I think my interpretation of the conversation on your question is it's maybe not necessarily about better or worse. It's just different tools for different purposes. And we may be able to use various real world data and evidence sources to maybe at least explore new hypotheses that may lead us in future directions. Mary.
Mary Short, former NICU nurse and industry representative. And one of Inc.'s goals is for a regulatory path forward for neonatal medicine. So my questions is for Dr. Kanto and Dr. Bax. With that in mind and having the Inc. database, will it be a necessary step or an important step to do a demonstration project like the FDA is doing to show the validity of this type of approach? And is that something that INC should consider as a next step?
I can go first. I think that what I've been struck by is how difficult it is to, other than with 20/20 hindsight, to see what the best action is at any one time. So I don't want to answer with a vague maybe, but if an opportunity arises for a demonstration project, by all means, take advantage of that. On the other hand, if there's data and evidence that suggests a new indication could be approved, it wouldn't be tomorrow in quotes, but next, without a demonstration project, that's fine. I'll point out one other guidance that I didn't include, not to focus too much on it, but there's a 2019, we call it shorthand substantial evidence of effectiveness guidance. It really lays out the approach that FDA takes. And when we say regulatory flexibility, we don't mean that we're lowering our standards. It means that we understand in given context with very small sample sizes that different tools have to be used. So I would say if you put together the available data, consider the current state of the art regarding study design, question is like, how did I'm not giving the sponsor a shout out, but how did the sponsor realize that there was an opportunity for prograph tacrolimus at that point in time? So I guess chance favors a prepared mind comes to mind as an idiom. So I think our job collectively, and actually outside of the regulatory agency, so euphemistic, your job is to look for opportunities where the data are ready for either a demonstration project to move it down the field, or an actual submission to take it across the goal line. I think that's the best I can give in terms of a detailed answer, is it ultimately is case by case, and I'll defer to my wonderful colleagues at FDA in this clinical space to be the first point of contact to sort of bounce ideas off of, if that helps.
I would actually respond in a similar way. So it's not a one-off. It's iterative. What I already mentioned is that we are learning and learning from case to case. So colleagues have been looking into examples in the adult space, and they have seen that only a couple of cases actually succeeded. So succeeded in showing that, for example, submitted real-world evidence was beneficial to regulatory decision-making in view of an authorization. But not so in other cases. So, and then we need to learn from these other cases. So I would also go for a similar approach and take it forward as it goes along, and then just confront us with it, and then we'll take it further. And obviously also, as we always say, and we also mean it, please come along and discuss and communicate during your development. So if we sit there together, we can work on it together. I mean, that is partially what we're doing already here, perhaps not in a case specific space, but also when it becomes concrete, please come and communicate.
Thank you, Mary. One more. Did you have a comment?
Yeah.
Yep.
Just moving away. About being a pharmacologist, PK, how the drug concentration changes with dose. In clinical trials, patients are fairly homogeneous, so we can get average information, but in the real world, patients are different. Different age groups, especially neonates, very mature, and also different renal functions. That kind of data we can only get in real world patient information.
Yep.
And I think there's a lot of effort in the US, and we're starting to do similar collaboration with industry, but that we really need real world data.
Fully agree.
Hi,
Jerry.
Jerry Bear from FDA. One comment and one question. John, to your, Davis, to your point about the trial that generated accelerated approval for the preterm birth drug, and then a large confirmatory trial, which was not confirmatory. In that case, there was also tremendous amount of real world evidence presented at that advisory committee, and the real world evidence also did not show an effect. So they used observational studies, gathered all the data possible to try to, figure out truth. And so the agency is willing to consider that information as well. A question for anyone really on the panel, because I believe that Sheila brought this up. There's tremendous site differences in outcomes. And presumably there are site differences in practices and populations. Is there some way that this real world data set that has been gathered can help to determine the underlying cause, or sort of smooth out all of that noise so that we can tell, what the truth is and what's working and what's not working?
I can't speak to the actual data set we have, but I know that the Vermont Oxford network uses the risk adjustment calculation for the smoothing. I don't know how good it is. I think it's been tested for a long time. I haven't looked at it recently, but I know that that's one strategy. Problem when you do that is that you also, in my estimation, is you mask what's actually going on in terms of the variation, and you potentially cover up inequity So that's my angle on it. I would love to see some better risk adjustment. And perhaps maybe that's something we could do today or in the next year to look and see what are these regional variations, what are these unit type variations. I remember I got my start in research because I was the data abstractor for our unit for Vermont Oxford. So I completed those surveys that were used for the risk adjustment. I think it was 4 pages long. So it was quite extensive.
Julie, do you agree that this has often been approached in terms of patient level, provider level, and then institutional level, characteristics, and that doesn't fully cover it either, because when you talk about patient level, there's clinical factors, there's also socio-demographic factors, and that spills over into regional differences as well. So it's a really challenging space, but going from little research or virtually no research to the best we can do, again, is part of the path forward to say that here's what was published that does try to get at, because I've seen reports of clinical trials. There's a distribution of outcomes by clinical trial site, but there it's more constricted because you have that protocol that's saying everyone should do it the same way. All bets are off based on your question, Jerry, right? In the real world data space. So again, another example linked to the other question of how it's harder to do quote unquote real world evidence the right way. But that doesn't mean we shouldn't try.
And I'm also looking forward, Jerry, to using the data set to really explore issues with health equity. I think in any one individual clinical trial, you can't possibly encompass all the different ethnic and racial diversity. I know people tried, certainly with the COVID vaccine trials. At the end, they sort of stopped admitting all the Caucasians and just waited to get other population involved. But I'm hoping can you look at that. One of the investigators working with me is looking at prenatal opioid exposure, something near and dear to both our hearts. And she's found a really tremendous difference in males versus females with respect to gene expression, feeding behaviors, and evidence of white matter injury at the time of birth by MRI. So again, that's not anything that anyone's really considered before. So again, I'm hoping we can use that data set to really explore some of these opportunities. And again, as John suggested, using it as a sequela and have as well, or an adjunct to clinical trials that do exist, or even more importantly, are being planned. So I'm hoping that we'll get people, investigators and sponsors coming to us and say, we have an important question I'd like to ask. What is the real world data and evidence out there that we can use to help design our trial better so that we're not looking at it the other way around?
In the sponsor's perspective, I certainly hope so. We have time for one more question. Bob.
Would you explain to me why RCTs and the control group in an RCT wouldn't provide normative data that would maybe be cleaner and more representative, I guess, of the real world than the real world data?
Well, actually, in the paper that we showed and cited, we do have a line in there. I didn't memorize it, but basically we emphasize that clinical trial participants exist and are analyzed in the real world. That is still, in one sense, real world data. The reason why a comparator arm from a randomized trial taken off the shelf might not be the best thing to use is because of in general, the restrictive inclusion-exclusion criteria. Regardless of how restrictive they are, you're applying that filter to say in the spirit of getting a highly valid, internally valid answer. If you go back far enough, right, oncology trials, sorry, I'm an internist, not a pediatrician. If you were over 65 and had any comorbidity, you were excluded from a randomized trial of chemotherapy. I started out my practice in the Department of Veterans Affairs. I didn't meet a single oncologist the New England Journal papers were nice, but that was useless because you couldn't give that dose. You would knock off the kidneys or the liver or both, et cetera. So we are supportive. There's a project being done where the control arms of randomized trials are being archived. They can be useful, they should be used, but with the caveat that they are probably on average going to be more restrictive. So therefore you have a higher bar to clear to say that it can be used as a comparator. Whereas the challenge for the observational study is less on the generalizability side, more on the validity side, because you have a heterogeneous population. How do you know you're comparing apples to apples with regard to outcome? Does that help?
Yes, indeed. Don,
Well, I was just going to say, there was a recent trial of buprenorphine compared to morphine for treatment of opioid withdrawal and neonates. And it was in the New England Journal. Everyone was very excited, and we're using it now to base future NIH trials on it. But when you looked at that morphine group, the control group, those kids, those outcomes were bad. So, and the buprenorphine group was better, but it was comparable to our morphine group using our real world data.
Gotcha.
So, based on that one small randomized controlled trial, people thought that buprenorphine has really made a significant impact. I wasn't certain whether it really did, or it's just that the control group, just by chance, with a small group of 30, 40 control infants, was just worse than usual.
And we're going to be continuing this conversation right after lunch, specifically on this topic, looking at various pros and cons of data sources, specifically around this question. So I would like to thank the panelists very much. I hope you all enjoyed the conversation. I know I certainly did. And we will be continuing on the same theme as we move right after lunch. So, Kamalja.
Thank you, Tom. And thank you so much to the panelists for such a stimulating discussion today. And, we did not really run out of the questions. We ran out of time here. So, we're going to go for a very needed break right now and be back at 12:30 p.m. Eastern. And now, finally, we are at the session that everybody has been waiting for. So no, seriously, because it's control arm is optimization such a big issue in many of the rare disease clinical trials, especially so for the neonatal clinical trials, given such a great difficulty sponsors have in many cases completing their clinical trials simply because there are not enough numbers available. In case of our other stakeholders like parents, they always worry about, why should they enroll their kid in a clinical trial if they're not going to get the active medication or active drug. So this is what we wanted to discuss in this session. The title is Control Arm Optimization for Neonatal Clinical Trials, and there are going to be a variety of issues discussed. Need for control untreated comparators for these clinical trials. Are there any other methods available that can actually reduce to some extent reliance on placebos, for example, in neonatal clinical trials? What sort of methodologies are there? What we can do better? And how, various regulators from FDA, EMA, and other stakeholders, industry, academia, parents actually think about this issue. So the presenters for this topic are Tom Miller from Bayer and Deb Dicenza, who's Inc.'s parent co-director. After this presentation, we're going to have a panel discussion of various subject matter experts and stakeholders who are working on this issue. So without further ado, I'm going to ask Tom Miller to come and provide his perspectives. Following that, it'll be Deb Dicenza.
My final trip to the podium for this meeting. But a topic that I am quite interested in, and I suspect a number that are attending this session today are as well. So maybe starting with a question, is it time to rethink our approach with studies intending to enroll neonates or a broader infant population? And why might that question even be asked? Maybe to start the conversation. I suspect many of us, and with whatever hat we have in our relevant constituency, could appreciate some of the longstanding challenges associated with study feasibility, enrollment fatigue, premature study closure. I did a quick scan of the literature in preparation for conversations today, with a specific interest in curiosity as to what proportion of trials that we initiate don't reach enrollment goals. And while the data was all over the map, I would say a reasonable range to contemplate as our reality today is somewhere in the order of magnitude of 20 to 40%. And you know the drill, what happens under these circumstances, right? We'll decide to break the blind. We're not powered to test the hypothesis, and the conclusion is more research is necessary. And for many of the sessions that I have, and I suspect many of you have attended at PAS for the last few decades, it's a very repeatable theme. So the question is, what if anything, might we be able to contemplate as an alternative approach? So why might we consider alternative study designs? Mike, in his great talk earlier today, made reference to one of the studies that my group had recently completed, and we were successful in employing an external control arm for determination of safety and efficacy. And one of the things that we noticed is that we had a relatively high ratio of informed consent approach to enrollment. When we were having a conversation with mom and dad or the caretakers, where we could indicate that the majority of children that would participate in the study had the potential to benefit from active if such a benefit exists. We were able to roll the study with incredible efficiency in this regard during the pandemic months, which was certainly no easy feat. The fact that we had fewer prospectively enrolled patients in the study equaled a faster cycle time for the study overall. This was a study in 120 infants roughly, that we enrolled in 25 countries in roughly the same number of centers in about 9 months. And it may improve our probability of technical success, getting to the finish line with the intended sample size to test the hypothesis. So as we open up the conversation, some things that I know I think about and my team thinks quite a bit about as we contemplate what could be. This concept of fit for purpose, which has been a little bit of a recurring theme so far in general for use of real world evidence. What are we using the data for? The sourcing of data is something that we touched on a few times today, and I promised Bob we'd come back to this very topic in this session. Is the data representative? Is it a contemporary representative current standard of care? That's one of the challenges. Some of these studies that we might be able to reference may have been conducted some time ago. And there's the question of whether or not care has changed. And is it representative of care in the modern age? And there are many sources where data can emanate from, right? Of course, the use of EMR data is the dream because it's so vast. There are millions and millions of data points in just a single country worth of potential data. But there's the question of quality control and how reliable is that data to point us to truth? And do the methodologies the curation methodologies that are available today allow us to think about utilizing such data versus, for example, using peer-reviewed aggregated data sets with contemporary representation of natural history? Or in circumstances along these lines, should we consider conducting our own curated natural history study? So we have as close to perfect representation of the intended patient population for subsequent Verum comparison. Data quality matters, and this has been enforced with many conversations that I've had with regulators at various health authorities around the world. And I'm very curious to get the rest of the panel's take on what good looks like in this regard. So with that, I will transition to Deb and ultimately to Klaus for the panel discussion. Thank you.
So no story this time, I'm sorry. But we are going to start with a video that was done by Betsy Pillon of Hope for HIE.
placebo optimization is really an interesting topic. And as far as, it goes with HIE, something that we're exploring and able to do is share lived parent experiences about what they prefer. And that is really aligned with the Corey model of research. And I'm excited that Corey has announced funding for a comparative effectiveness study for mild HIE in particular. We know that, the different methodologies of these studies really matters when it comes to enrolling families, and that, the standard has been random controlled, but the communication is really critically important of how this is done with consent forms, with just educating families on, risk assessment, on benefits, and how they can, how others have contributed to the science before them, and how they can be a part of the science moving forward that can benefit not only their children, but also those to come. It is through that we can continue to move forward these efforts and make sure that families have a halo of support around them, not only to help them understand in these very tenuous times when you're asking people to opt into these trials, but also have that support throughout any trial, timeline, partnering with patient advocacy organizations, such as those that are involved with INC, is critically important. One of the benefits is we can definitely help with decreasing attrition, continuing communication, and helping, again, develop that halo of support around those that are involved in these. So thank you for everyone who is participating in this session, and for the advancement in considering how families might feel about receiving a placebo and optimizing trials accordingly.
Thank you. She pointed out the halo of support, which I love because I spoke to it earlier today, but she put it so succinctly. As a parent in this space, I am aware of the challenges to recruit patients in a highly stressful and fragile situation. I am aware that data of all types can help where recruitment cannot. With this in mind, it is important to me that any type of synthetic control has the same regulatory rigor as a typical clinical trial. Parents need to be aware of how this is different from a regular clinical trial and what the risks and benefits are. And to the trial, but more importantly to the patient. We also need to make sure that whatever therapies that come from such research are truly representative of what a control should be. How does it reflect reality? We also need to make sure that whatever data is used, we can also accept its flawed ability to represent a real framework of a trial. These are patient lives we are working with, and there is often little room for error. I will end with this before I read Kaori's statement. What would you want to be done for your loved one? Kaori from Japan wrote a wonderful statement. I want to thank Wakako for helping lift her up. As a parent, definitely, it would make me cringe and agonize over the decision of whether or not to enroll my infant in any study, especially if I understood fully that in a randomized clinical trial, which is the highest scientific standard today, only 1/2 of the enrolled babies would receive the actual new treatment versus the traditional therapy or no therapy. I may opt out of my baby enrolling simply because of mental stress and knowing that my son may not benefit from the potentially good and new therapy at all. These concepts are not easy for us parents to easily understand or support. If the placebo group can be created using the already existing data of average NICU babies based on a large database to serve as a placebo group, my thoughts as a mom of an extremely low birth weight baby, that I would definitely find this type of study more attractive. For those who are listening, I would ask that you would kindly consider the moms and dads' feelings when these study opportunities are presented to us for the enrollment of our precious treasure, our babies. Sure, intellectually, we want to contribute to science, but our babies are so special that our intellectual understanding is not what often guides our decision to enroll or not enroll our babies. I hope these personal, candid thoughts are helpful to you as you approach the innovation in a study design. Thank you.
Okay. So, let's start with a couple of things, and I'll use my moderator prerogative to switch the title of the session to control arm optimization, as we discussed. And then I will also take a minute to also address one of the themes of a couple of questions that we heard in the previous session. What demonstration projects could we think of? I want to remind everybody that we have 2 of them already ongoing with the real world data, looking at the lab value standards for NICU usages and the bronchopulmonary dysplasia disease progression model, but we shouldn't stop there. And I think this session is also going to provide an opportunity to tie things together. So let's start with addressing one of the elephants in the room. Why do we even need a control arm in clinical trials in the first place? So, as our statistician from a regulatory agency, can I pick on you first?
Absolutely. I think this is on. Is it on?
Yes.
Okay, great. Thanks. You know, control arms generally serve the purpose of us being able to look at an experimental arm and be able to say, yes, we know that what we're seeing in this experimental arm is attributable to the drug itself. So in a randomized control study, the goal is that randomly assign patients to either arm, and that helps us know that at the end of the day, any difference in treatment effect or any difference in the outcome that you're measuring is attributable to the drug or intervention of interest. Now, there's so many situations where randomization is just not possible. Either it's not ethical, or there's an unwillingness of the community, the patient community, the investigator community to randomize. I should note that I am a statistician. I work for the FDA, and my area of expertise aligns mostly with rare and pediatric cancer. So we do encounter this quite a bit. So there are often single arm studies that you can do if you have a very specific objective measure of efficacy. So for example, in oncology, that's usually tumor response rate. Is your tumor shrinking enough that we can say, yes, this drug is helping you? Because we know that for almost all cancers, in the absence of an intervention, a tumor is not going to shrink on its own. So in other disease areas, this is also applicable, but sometimes it's not quite enough. And in oncology, we have this often as well, that, you know, you have tumors that have some spontaneous regression, or they're extremely slow growing tumors. And so it's very hard to see if there's enough of a response to really translate into clinical benefit. In those cases, sometimes it's beneficial to look at external data to help us understand whether or not what we're seeing is really a benefit to the patients that's attributable to the intervention of interest. And so that's where we get the concept of external controls, which are also sometimes called synthetic controls. Synthetic controls could also mean completely synthetic data, simulated data, whereas external controls is a more broad term indicating any sort of data that you would use external from the clinical study.
Beautifully stated. So, Ralph, a lot of times we hear misconceptions to the tune of, oh, FDA and EMA don't think the same way, and they have differences of opinion and this and that. But as we were discussing with you prepping for this session, it became pretty clear that once you peel the onion, it's all about the scientific rigor and how that's the common denominator, and there's no difference there. It's just the notion of, as stewards of public health, the role that you need to fill within the regulations that govern your agency. So do you want to comment from the EMA perspective on the notion of being innovative with the setup and the design of these studies?
Yes, of course. And I'm glad you started with my statistician colleague, because, you know, my former life was also in the clinical field and actually in neonatology. So I don't understand anything about statistics at all. So I was, you know, more the lower grade of neonatologist potentially. Yes, but what we in the end want is not exactly, we're not asking for a placebo to ask for a placebo, as we want to understand and find a way of discriminating that really this drug is having an effect, having a treatment effect, and not any other effects having a bias. So how can we achieve this? And if I hear, and we have been confronted a lot also in our committee when we have patient representative interactions, that we are asked, why are you actually preventing us from getting this medicine? Why are you putting up such a hurdle? And if I hear that parents then say, I might be denied such a treatment with a new medicinal product or investigational medicinal product, we have to ask also, there could be a harm giving it. So you might be even denied the harm of it. I know it's a difficult concept, because I know, and also being a parent myself, we put our hope into, and specifically also in the pediatric oncology and also in the neonatology field, into any new medicine. And please give me this any new medicine. So in the end, we should think about how can we discriminate these different effects, and it doesn't have to be placebo. In Europe, we have been using a lot of active controls, actually, where we then sometimes have the difficulties that this active control, so giving an alternative medicine, whether this is authorized or not, and often it won't be authorized. It will be, might be standard of care, as you know, but not authorized. But even here, we do see some flexibility. And to go on to your question about external controls here, exactly, it's the same case. Can this external control or historical control help us in discriminating? And that will be the question. It reminds me of the situation when we discussed clinical trials in the HIV field, actually. And at the time, 8 years ago or so, in Eastern Europe, brain cooling was not standard of care. So so the applicant designed 2 sets of studies, one with brain cooling and one without. So just to show you how, if we now use this as historical control, non-brain cooled children might then not be, now it's the standard of care everywhere, might not give us the same comparative measure, just as a practical example. So it is about the methodology and not whether it has to be placebo or not.
Right. Exactly. So right there, I think we've heard one important take-home message. If we want to demonstrate efficacy and safety for a novel medical product, there needs to be a comparison against the notion of the untreated population or the population that is treated with the quote unquote standard of care versus the new medical product for both efficacy and safety. And that's one of the points that Ralph highlighted. But that brings us into another take-home message, and I'll pick on Tom next, because you're driving product development, right? And one of the key aspects in bringing that point home has to deal with what question are you intending to answer with a specific design at a specific stage in the development process, and how you think about those aspects from an industry perspective when you're faced with the challenge of designing these studies. Can you comment on that?
I will do my best. So in our last session, the panel made reference to a couple of different applications or potential applications where real world data and derivatively real world evidence may play a useful role. One of the areas that I've been particularly interested in as of late is particularly for these rare pediatric diseases, including diseases of infancy, that we've made reference to for some time. It very clearly appears in many circumstances that there are many subordinate endotypes of disease, and how a biomarker of relevance to understand disease progression over time may change differently in one endotype or variant of disease versus another endotype or variant of disease. And I suspect this audience will appreciate more than most that the literature is not often replete with information to guide us. So I think one of the topics that I know our team has been interested in is, sometimes maybe the best way to get at the answers to these questions is to create the data ourselves in the context, for example, of a natural history study, where it may allow us to look at different cross sections or cohorts of disease before we even think about intervention with a test article, so we can provide evidence to other important stakeholders in our ecosystem, including regulators, that we now have a good sense of how disease progresses as measured by biomarker X or Y or Z. And this gives us rationale to say, and now we could have maybe a bit more of an intelligent conversation about how to design the study and what inclusion and exclusion criteria should look like. Of course, back to your modified title that we put in place, we are also very much interested in the topic of alternative control strategies. In some respects, due to some of the comments that Deb made and some of the comments that I made as well, and there's a practical side of doing the studies, and that's getting the studies done. So how do we find that right balance between feasibility and appropriate scientific rigor to answer the question in front of us? I think is something that we're looking to try to find a good Venn diagram with all stakeholders in our ecosystem.
Perfect. And that's right there, another key take-home message from this discussion, making sure that it is understood that our regulatory colleagues are open to have conversations, as you always remind everybody, often and early, to have that kind of a discussion around the importance of the different aspects of a design, keeping in mind that you can think of specific ways to set up the comparator arm to make sure that you can demonstrate efficacy versus safety. But with that, we have two people here that are in the trenches, in the research space, actually making things happen with interacting directly with the patients. And so I'm going to turn to Hide next, because I'd like you to comment on one of the observations you made in the last session, the quality of the data, where we were discussing the fit for purposeness of the data, or the fitness for purpose, and how that needs to depend on the question. It is not a one size fits all answer, that the quality is universal, and how you have instituted efforts to ensure that progressively the data can give us the answers to the question that Tom put on the table, how can we better understand the disease trajectory so that we understand the untreated populations, so that we can understand how to best optimize the control arms? Can you comment on your efforts on that front?
Ooh, very difficult question. To set up a good control, we need to understand, like Tom mentioned, we need to understand the natural history, how biomarkers change over time, this kind of information. Not in neonatology, but from my experience in another area, especially for rare disease, doctor says different things because they see very few patients each. And in one study, they said, okay, well, all patients get liver transplant in a couple of years. And I didn't believe them. I did national survey and turned out that, yes, there are some patients who get very worse quickly, but there are other patients who start to have liver cirrhosis very slowly. And so I told them to set up a new endpoint, that liver pathophysiology. So we had a group of pathologists who set up a score, and we branded them, showed them tissue samples, and see if it's really that there's no difference between evaluators. We saw some variability, so we asked them second time to modify the score, and then we gave them blind sample again to make sure it works. Then we went to clinical trial. So it's not easy to just use electronic health record to get the right answer. But it's important to go to each patient very carefully and then decide what can be control, what can be historical control, what the best biomarker is, and then go ahead. And especially in the area where we have very few experiences, we may have to find out a new biomarker or a new score.
Great. And that's another important take-home message, right? Because it links to understanding exactly the question that you want to answer and how you set course to inform the data gathering to answer that question. But Sheila, at the U of A, you're exposed to some really relevant populations that tend to not necessarily be paid attention to as much. So another comment that you made in the previous session was about the notion of what you know intrinsically from the nursing community that makes the most sense to facilitate those data gathering exercises, especially with those communities. Can you comment on how to weave in that thinking as we continuously think about optimizing not only the control arms, but also accessibility to research and clinical trials?
Wow, that's a big one. I kind of feel like the person on the Jeopardy game at the very end. Okay. Okay.
Wait till I get to Deb.
Oh gosh. Wow, Deb, get ready. So I think what you're asking me has to do with completeness of data, engaging all the communities, and there was some other feature.
Accessibility.
So if I could just come at this the back way. So one of the things I think is an interesting thing to think of when you talk about placebo optimization is that typically we think of a one-to-one. Like we have two potential participants, we randomize one to the control arm and we randomize the other to the experimental. So that's the typical way of thinking of it. Recently, I was in a project where we were looking at how we could optimize because we had a very potentially beneficial treatment for necrotizing enterocolitis. And necrotizing enterocolitis is rare. And so we needed to figure out how we could maximize our potential population, minimize our sites, or at least optimize our sites. And so one of the things that was brought up is what if we did a 2 or 3 to 1 match, which was a different way of looking at optimization, maybe a little different from what we're talking about completely right now. But it's one of the options on the table. So in terms of completeness of data, I think that's something you can structure. You can either structure on entry or structure on access for the data completeness. And that's I would assume that in a clinical trial context, you're very structured in what you'd be kind of collecting. And so you could work with clinical research coordinators, which are often nurses and others, to make sure that that data is complete. In terms of accessibility of the data, is that the next question?
Accessibility to research.
To research. Okay, so that piece of it, I'm going to also come at a different direction, if that's okay. Because I think that there might be an inherent issue here that I think we need to probably talk about, which is how acceptable is this to participants to enroll in a trial like this, and how clear is it for them to understand and say yes to a consenting conversation. So, so in terms of accessibility, I define that a little bit differently, but I'll come back to how you define it. So the accessibility issue in terms of when you mentioned earlier, this issue of you want the benefit of the drug, but you also, I'm a bit risk averse myself. So I want to know that I'm not bearing all of the responsibility for the risk. So if I have a baby in the NICU and somebody's bringing me a study where we're only primarily enrolling treatment group and we're getting our control arm from somewhere else, I'm thinking, well, how are they going to find out if this is dangerous to my baby? So I understand that there are good answers to that question. I think that's just part of the conversation that has to be had. So in terms of accessibility, and when you approach underrepresented groups for research participation, which are already going to be somewhat risk averse and questioning your motives because of the historical context that they come from, you need to be even more obvious about that conversation and say, you're not bearing, you want, that population is worried that they're bearing all the risk of study participation. And so in Southern Arizona, I work with people in a NICU close by, which are 60% Hispanic, and many come from our sister units across the border. They get flown over or ambulanced over because they need care. And so in that context, in approaching an already worried family, you have to be extra careful in how you communicate that, but they would also potentially benefit. So in terms of data accessibility, is that what your question was? You're talking about how could you actually get to that data?
Right.
So that is something that our informatics colleagues could help us work through. Yeah.
Yes, Deb, you want to comment right away? Yes.
Thanks for letting me go in the back door and walk back and forth in the kitchen a little bit before I answer that question.
That was nicely done.
Okay, couple of points. Obviously, education is a key component, absolute key, and how you talk to the patients. And that is, and I know we're all about literacy levels and everything else, but I'm a co-founder of the Alliance for Black NICU Families, a nonprofit. And in talking to these families, you don't talk down to them, talk to them. And in terms of health equity, which Sheila spoke to previously, I mean, this can be couched in that way, equal access. It's equal access, but it has to have that same rigor that you have been coaching me about. So I'm saying rigor, rigor, rigor. But if you have that and you can provide equal access, then there's an acceptability there. You can build some trust.
And I want you to keep elaborating on that point, because also in preparing for this panel, we talked about the important notion of the two-way street in community. You talked about, you talk with them, but you also listen to them a lot.
Yes.
And that has to do with one key aspect that is very important to our regulatory colleagues and our industry colleagues, patient-focused drug development.
Yes.
And how this is special in the neonatal population.
Okay, right. Yes. As you had spoken to Katie yesterday when we all met up, talked about it's a two-way street listening and working together, you help create that environment of scientific accessibility. And I just think it's really, you work together. I always talk about it being a partnership in the NICU. It's the same thing here. You have to partner together. It's not one side getting something from the other. It's you working together, create that environment so everybody wins.
Yes. And I want to bring it, yes, question, yes, please.
When we were in the NICU, we're enrolled in a drug study. And the most profound moments in that was our neonatologist came to my husband and to me, sat down eyeball to eyeball. We had already established deep trust in Dr. Wagonka. Anything he said, go. And I think in a lot of ways, that profound of knowing that he had our best interest in, he was deeply vested in our family, already established this connection. How do you feel that in enrolling family to this into studies?
Right. And so, do you want to comment on that one, Deb?
Exactly. I mean, but not every unit's going to have that kind of opportunity to do that. And you have some scenarios in certain trials, I'm sure, where it's an emergency. You don't have that kind of discussion. So it definitely, I think when you speak to the culture in a NICU and being having empathy and all that, that's great. But when an emergency arises is when you get tested on that. And if you have to enroll in a clinical trial, how do you do that efficiently with empathy? It's a fine line, my opinion. It's hard. And I know that's what you work on, which is great.
Right. But that actually brings up a really important point. The dialogue, it's important that the dialogue happens in the trenches with the families in the NICU, but it can't stop there. It has to be an ongoing dialogue all the time. But I'll hold that elaboration for this question.
No, I don't hear the one from Bayern. No, maybe more directly a comment to that and to what you said. Our experience is we had a treatment where the treatment decision had to be taken within 48 hours and from the diagnosis to the treatment. So what we tried to do in that way, because at least in Europe and in a lot of developed worlds, preterm labor doesn't fall from the sky. Normally, women are also in care or in a hospital or anywhere well before. So we tried to provide more generic information on what happens with a preterm, what could you expect as effects, as bad effects, and obviously the indication in question was one of the possible worst outcomes. So a little bit prepare sensitivity upfront, which obviously helped to the recruitment and helped the investigator or the doctor, the treating physician, to start the discussion on a possible trial with that parents in that very, very short period of time. He had to decide if he does standard of care or if he does a trial treatment.
Right. That was a great, yes.
So
Mark,
I think companies
please.
can do a lot for this.
Yes.
So, Mark Turner, coming to the concept of optimizing the comparator arm, I think there's a lot of work to do to educate the average physician and nurse to build that trust, because the training that we've all had is that you compare the two groups and they're balanced in theory, and therefore we can draw a conclusion. But I think the ways that people are talking here will need to re-educate people so they can continue to offer that support that is so essential. And I think that needs to be part of any future work. It's not just outlining some nice, elegant regulatory or mathematical or real world process. It's about selling that story and developing that narrative that makes sense to everybody else in the room, because otherwise the trust goes. And if you're saying here's a new medicine, there aren't any comparators, then some people might find that a little bit uncomfortable. So you need to be able to build in the theory and the practice for that study. How are you going to make sure that there are no adverse effects unless you've got that comparison? So yeah, I think education for lots of people is part of the picture.
Yes, but the educational part needs to be linked, and I love that comment because it needs to be linked to action and examples. And one of the areas that I think we need to embrace and kind of tackle head on is this notion of another thing that we hear from the ultimate owners of the data, and that's you guys, the patients. And we are all patients at one point in our lives. And what is the one cry that I have heard in 29 therapeutic areas that we work on, and even the ones that we haven't tackled yet, from the patients? When they recognize the power that their data can have, they tell us unequivocally, we want our data to have maximum impact beyond the primary analysis of a trial. And I think that represents a really great opportunity because it links with what was being said about optimizing prospective data collection exercises, regardless if in a formal research environment or not, and linking that to specific questions, the data sharing and accessibility piece, and then linking that to quantitative analyses of disease progression, disease dynamics, because there's a comment in the chat about sources of variability. And I want to bring it back to the statistics in the equation. I mean, would you agree that ultimately one of the components of the game is being able to identify and quantify and thus handle sources of variability to optimize the design of trials? And that goes for the active arm as well as the control arms.
Absolutely. In the randomized trial setting, we have the benefit of hoping and believing and assuming that the variability is same across the arms. You're randomizing across centers. You may have patients at different centers who have slightly different experiences, but you'll have patients on both arms from those centers generally. And so, there are things we can do prospectively in those studies to control for that variability. And then also we hope that randomization is, if you have a large enough trial, is going to kind of wipe out that variability such that what you're left with is the treatment effect. When you're comparing two different sources of data, which is what you're doing when you're incorporating an external control rather than a concurrent or a control arm within a single study, the data may be extremely variable for various reasons. So like the first reason, and the one that we're generally the most concerned about, is that just the underlying populations are drastically different. So I'll give an example of this. In a clinical trial, most of the time we're enrolling patients at academic medical centers, and your patient population at those centers may be different than the patients in like community hospitals. And then if you were to compare that clinical trial data to, let's say, data collected in a registry that was mostly done at smaller community hospitals, then we wouldn't really know, despite collecting lots of variables that describe their demographic and disease and clinical characteristics. It would be very hard for us to say, well, at the end of the day, that these are the same two groups of patients. So you can minimize some of that variability. There are other forms, by the way. I don't want to bore you in the sense of that there are a lot of really great guidance documents from the FDA that describe these in detail, things that you should look out for in terms of variability. But I think what's most important is to think about what type of data is, and I know you guys have heard this word today, fit for purpose, but what is the appropriate data to compare to? That's really what it comes down to, is that, maybe a registry is an appropriate data set to compare to, because honestly, the setting of care doesn't matter that much. Maybe the best thing to do is to look for another clinical trial that has the standard of care that you were looking for, either as the control arm or it was the experimental arm in that trial, and that's how it got established as the standard of care. There are a lot of different opportunities with prior data that are out there that may be appropriate for your particular setting, but it comes down to what is the question that you are asking in your clinical trial and what data can help you answer that question. The biggest thing that I see where studies fail when they're using external data, they try to change the question for the data that's available. And the problem with that is that may not be a clinically meaningful question.
Beautiful. Clinical meaningfulness and the fitness of purpose of the data for the question. Nick, you live in the trenches with one key component of this, the data diagnosis for that fitness of purpose. I want you to comment on the experiences you've gathered in dealing with the ink data.
Yeah. So just as a general statement, what I would say is the whole process of taking the data that's available to you and generating a synthetic control arm is this iterative process where you go back and forth between the question you're trying to answer and the data and trying to ask, do we have something in our database that's actually fit for this purpose? And, does it satisfy our quality criteria, which has been mentioned several times. And just because there is, high quality data at your disposal doesn't mean it's necessarily fit for purpose. It doesn't mean that, the quality doesn't always correlate with the usefulness of that data for the question that you're trying to answer. So, and as far as the specific case of the INC data, what I would say is that we have had a lot of variability. A lot of the data that we have is very high quality. It is seems to be rigorously gathered. Our data team has done an excellent job of processing and keeping track of all of the different sources of error and sources of variability. And then they warn people on the quantitative side, like myself, to say, hey, keep an eye out for this potential source of error. And then we can incorporate that into our statistical modeling strategies. So what I would say is that kind of the end goal, if you look 10, 20, 30 years down the line, what I think we would want to see is somebody is designing a trial. They want potentially to use a synthetic control population. They don't know how feasible that is. They would turn to a database like INC and say, hey, do you have a sufficient quantity, sufficient quality, sufficient relevance of data to build a synthetic population for us? And then we could come back, theoretically quite rapidly and say, yes, absolutely, here's a synthetic population. That synthetic population might consist of real patients from the database, but it also might consist of synthetically generated patients using more advanced methodologies that are still in the process of being verified. And going back to another comment that was made earlier, I think that all of this, all of the usage of any of these tools, right? They're all just different new tools, has a lot to do with the trust and the verification and validation of them, and saying, we have this tool, it's new, it's different, but you can trust it because we have done this, long-term verification process, validation process, to make sure that the methodologies that generate those patient populations, the synthetic patients, are high quality and fit for purpose in whatever setting. So
exactly. And that really brings everything home, right? Because it's you mentioned that we probably couldn't say things publicly. Now you've heard the public statement. That we have been very pleasantly surprised with the level of rigor with which the EHR data that we have received has been collected. We were bracing, I'm not going to lie. We were bracing for, we're going to get into this and we're going to get slapped in the face really quickly. That has not been the case. But Jerry, please go ahead with your question.
No, you sounded like you were mid-sentence, Klaus.
I'm going to hold that thought because I want to hear you out.
Jerry Bear, FDA. Like Ralph, I'm not smart enough to be a statistician, and I'm also not a data scientist. However, I do have some questions. Obviously not everything is necessarily going to need to be a double-blind, randomized, controlled, placebo-controlled clinical trial. But if you're talking about completely supplanting a control group, what do you do to mitigate an open label study and the bias that may be created by, giving everybody the treatment and everyone thinks it works and yay, and then we're going to compare it to something completely external? I don't know who I'm asking that question to, but help me understand.
Yes.
I can take a shot at it, but I welcome my fellow panelists to add to it, please. That's a very good question. I think It really depends on how objective your endpoint is, right? So if you have lab values as an endpoint, it's very difficult to say, oh, this lab value shows it's working if it's not, because you've got numbers. That being said, there are a lot of endpoints where there are subjective measures. Again, I unfortunately I'm not familiar with a lot of the trial designs or the specific endpoints you would use for neonatals. But in oncology, for example, tumor response is based on the assessor. Someone has to look at those scans and say, yep, I think it's now shrunk this much, and now I'm measuring it. And how do you make sure that that measurement isn't subject to bias? There are ways to kind of to reduce that risk. Generally, what we ask for is blinded independent review committee. So in oncology, and I'm sure that there is something similar in this specific disease area. I unfortunately can't tell you exactly what, but I've had people ask me before, well, but everyone's getting the drug. Won't the blinded independent review committee know that? No, they won't. They just know that all this data is coming from a single study. They don't know that it's a single arm study. You actually not hide, but you don't tell them that information. So they're truly just looking at each individual patient. Now, that does open the door to, well, what about the control arm? And the question is, how do you compare that to a control arm if you have really high quality assessment of your experimental arm? Well, the answer is that you need to have similar assessments of your control arm. So then you need to get for oncology, the radiologic scans, submit those to a blinded independent review committee and have them review those. Now, if you're comparing two clinical trials, this may not be an issue, but if you're comparing data from, let's say, a patient registry, maybe a lot harder. And in which case we may start looking at other objective endpoints that we know can be measured by both groups without this concern for bias. Usually for oncology, unfortunately, that's overall survival. But I think any disease where you can look at lab-based values, you have a much better opportunity for unbiased assessment.
So just a follow-up for the group, which is there's assessment, there's endpoints, but there's also variability in the treatment and in the way that patients might be treated, knowing that they're in a trial. And we all know that there's a lot of treatment variability in general in neonatology. So that's another part where I am concerned about bias.
Right. And bias is a form of what I would characterize as the bad variability, because then there's good variability where you want to capture that heterogeneity of the population that you can then, through the power of numbers and close to 300,000 or more individual patient records, is pretty darn good numbers to start looking at those sources of variability and start quantifying them and identifying them and putting them in the right context. And in that way, you can start mapping out, how does this have an effect on the disease dynamics? And how can I use that to optimize the design of the study for both the active and the control arm, whether or not it's going to be an innovative control or not? Tom.
Yeah, I think that was a great answer. And how quantifiable an endpoint is, I think, really matters in maybe ultimately concluding that we could put a check in the pro column. But conversely, I know another area of interest of this audience is neonatal pain. And I would say pain would be, in my mind, at the complete other end of the spectrum, which is a very subjective endpoint. And we know through countless adult clinical trials that there is a potential for a massive placebo effect. So I would not recommend applying methodologies along the lines that we're discussing here for those type of endpoints that have a bit more subjectivity to them. And pain is a very practical one for this audience.
Yes, but I'm gonna pick on Tom one minute, because what about the future? What about those regenerative treatments and how we think about those, because you live that in the trenches as well.
Yeah, so we touched upon a few different methodological approaches towards sourcing real world data. One of the areas that I've seen quite a bit of uptick of operational implementation with is with natural history studies relatively early in the development cycle, and perhaps years before any consideration for verum exposure might be contemplated. Sheila, you brought up the safety question, and I didn't mention this earlier, but I think one of the other very potentially useful aspects of natural history studies, oftentimes for a particular adverse event in a rare disease population, pediatric or otherwise, we may not even know what the expected background rate of an adverse event might be. So I think prospective conduct of a natural history study may give us some information that we could then subsequently apply to the test article or verum study, when ultimately the investigator or the sub-investigators have to make the determination as to whether or not, yes, an adverse event occurred, but is it attributable to drug, or is this within the expected range that we might anticipate because we did the homework in advance?
Right. And single source of data a lot of times don't provide an answer. That's why we need to integrate as many data sources as we can. Yes, question.
This is just a follow-up of Jerry's concerns, and one of them addresses the issue of some of these protocols have a very poor equipoise, so that you cannot get people to enroll. The neonatal faculty just don't want to do anything about that protocol. And we have to be really, we had to really look honestly at what was the problem with this particular protocol. It was a comparison between two drugs to close ductus arteriosus. And the faculty already felt that one drug was better than the other. So right at the very beginning, that protocol was already dead. So we have to really look at some of these when we are having a very poor enrolling protocol. We really have to look at the protocol and see what the problems are. And we have to try either to resolve it or to can it, because that was really quite a lesson that we learned after all the amount of educational programs that we were trying to do to get a buy-in. It was very clear that there was already very bad equipoise right at the beginning.
Right. And get the input from, again, the patients ultimately.
Right. So it doesn't matter what a good outcome measure you have, you know, like a cardiac echo, you know, measuring it and all of that stuff. They won't really, they try to avoid enrolling in that protocol.
Yeah. And so if the protocol is not fit to answer the question at hand, and it's not feasible to execute with the patients, and it doesn't answer something that is clinically meaningful to the patients, then of course you have to take a pass on those. But Ralph, I want to go back to another thing that you mentioned, and the efforts that we're seeing in globally, but certainly EMA and the EU has been very much invested in efforts to start looking at data sharing, data integration, and how to leverage that to inform and optimize clinical trial design. And I know that we have spent many hours speaking on the phone, but can you comment on opportunities that you see truly globally to start getting into the consideration of global data integration to really treat those geographical differences in standard of care as another quantifiable source of variability that we can address?
You might be, I mean, I'm glad that you are so optimistic. I'm also an optimistic person, but I think we have to as I alluded to in the European space, the difficulties we are facing, and which obviously you have also faced and potentially overcome, be it on an operational or methodological level. So here we are, very humble at the moment. We are seeing at the moment, or trying to observe in a way what is happening, and analyzing, encouraging on one hand and analyzing at the same time on the other hand, and seeing also quite a number of failures, which is not negative, because I mean that is we all have to learn from it, and that is why I mentioned just come forward. With sharing data, there's another hurdle in Europe, and I'm just mentioning GDPR, or data protection. I'm mentioning a PDCO member who is director of a children's hospital, who says he's not allowed to share anything just from I mean, as we have heard. So so many discussions we have at the moment in breakout sessions and so on are frustrating. But on the other hand, we have also discussions with legal colleagues who explain to me, and that is even more complicated than any statistic, explain to me that it is possible to share data even in Europe. And honestly, I mean, one of my aims was when I looked at your initiative, that it is really my frustration also. I mean, thankfully, we do have European data in your database, but it could be better, and we need to work out how to do this. We are potentially using, I mean, we are using in Europe, different vehicles and different infrastructure, because the whole setup is completely different. But in the end, it boils down to that we have to have platforms, and that we have to, in the end, it converges, and that is where I'm optimistic, and I hope we can also learn from your experience, and that we then can feed back into our discussions in the regulatory network, where I believe that seeing what is happening here, that neonatology and this space is one step ahead. So perhaps we could, as you also mentioned, perhaps we could use this really also to educate regulators, because as Jerry has mentioned, and we have to be also realistic and open about this. I mean, it is trust, and stakeholders and developers will have to, and I think also in a certain way, rightly so, convince regulators who are, let's say, put it this way, conservatively innovative. Because
you're an optimist too with that comment.
Yeah, but I think, and that has also some purpose, but sometimes we need to make some extra effort to do this, and I think it's worth it. And I'm just trying to come back to what we are looking for, and there are many ways, but to addressing the right way, and that is which is a common denominator. Thank you.
Right. So you touched on a really important, another take-home message, the learn and confirm paradigm, which when you do drug development right, that needs to be at the center of things. We can expand that notion to everything. and learn from the data, from the gaps in the data, to make sure that for prospective data generation, we have colleagues from NICHC and other places that actually fund research. That could be also a component of the conversation, and integrate everything. Tamara, please.
Hi, Tamara Lewis from Sick Kids in Toronto. So this has been a very helpful conversation, but I want to ask a very kind of pragmatic question. So we know that there are really rapid secular shifts in NICU management, meaning a cohort even from 5 years ago may have been exposed to a very different set of interventions than a current cohort. And so my question is about the INC real world data that's been collected is very important, and it's a great step in the right direction. But I think if you really want a very contemporary cohort that, let's say, a specific set of infants all affected by a certain disease process, has INC started the conversations around rapid iterations of creating these control arms or these external arms, and how INC can facilitate industry working with your academic partners? You're going to need very, very detailed EHR level data to do these external control arms. And if INC can be kind of a facilitator of that rapid collaboration to create contemporary cohorts.
Do you want to take that, Tom?
I'll try. So great question. Also sounds like a seed planted for a future working group on the chance you're interested in volunteering. I think we are still in very early innings, and I'm looking forward to the discussions later in this venue when we've been so focused on amassing the data set over the past couple of years. We haven't yet had the opportunity to kind of gauge any insights from that data set yet. So I think we have to learn to walk before we run a little bit. But I think the general approach that we're taking, where we view this not as a project that ends, but a project that can endure over time, with the goal of assuring that it represents a contemporary snapshot of modern day neonatal practice, I think at least preserves the option to ask those questions. But I think we have to take this first step first. Is that fair?
Absolutely fair. Yes.
Yeah, I was just maybe I think thank you, Michael, for the, talking about the European situation, and maybe to paint a little bit of hope into it, not for tomorrow, but maybe in some years. I think with the creation and the provision of funds for the European data space, we also see now discussions on the public-private front, so into the innovative health initiative, where we have discussions on public-private consortia, where also the European arm of C-Path is involved to create and to make data better accessible, and to come to European-wide approaches and the way to access data. But as we know, Europe, that will take a while, so that is not tomorrow, that is maybe the day after tomorrow.
Maybe, but I'm still an optimist, and based on what I've seen, I mean, because I'm going to be very blunt here. When we started this, we were told, you will never get data from Japan. Are you kidding? Are we kidding? You will never get data from Europe. Are we serious about this? It can happen. And we leveraged what we had before. We learned in the beginning from the adults, the classic way. But mind you that we had never had EHR data integration with clinical trial data. That had never happened. And in one pass, in 1 year, we moved from 0 to over 300,000 data points. That is an unbelievable testament to this collaboration. And so, Mark, you were in the trenches with us to make that happen.
Well, yeah, I think Professor Modi has a lot to participate from the UK. I think coming back to Tamara's point, for me, it's not just about numbers. Again, I'm a ignorant neonatologist. I think the social processes need to be built in as well, because it's about giving incentives and drivers to institutions and to sites and to individuals to provide the high quality data. Benchmarking is part of it, and maintaining the trust of the families as potential participants. So as as Tom and Tamara build into that workshop, that work group, I think it's important about those social processes, because without them, then the numbers aren't going to have the sufficient quality.
Anybody want to react to that?
Maybe just a quick one. I suspect we'd all have had experiences where, success begets enthusiasm and then subsequent success. So I'm hopeful that as we start our initial phases of our number crunching and see what insights we can glean from this, that will be a good motivator for centers to start to understand, well, hey, how can I use this for our own purposes? And then start that social exchange a bit more holistically.
Yes, and never losing track of the patient involvement with a lot of questions that we can start. Also, not just because EMA has a question or FDA has a question or industry has a question or our academic colleagues have a question. You guys must have a million questions. And those are the ones that start with the true clinical meaningfulness at the center. So just as you have been part of this journey, we really need you to be part of the journey for what comes next.
I mean, yes, absolutely. And again, it goes back to education and speaking to people with respect.
And listening to them.
It's the same seeds. Repeating.
Sheila.
Could I speak to this though? I think that there is an issue if we always go straight to education, because there is a person delivering that education in a neonatal context. And that person is highly variable, because you're not really, if you're creating a clinical trial, you can hire people to be your research coordinators and to run the thing and make it work. But once this goes into actual, broad scale use, or even trying to do big trials, you have a person who maybe, our colleague mentioned how the neonatologist wouldn't enroll in that one trial because they didn't believe that one drug was worse than another. There was an issue there. And so I think there's an opportunity for a group like this to at least document what the acceptability and the gaps are in actually bridging this, because eventually, once you create this beautiful real world evidence, you're going to want clinicians to go ahead and adopt it. So I think that there's some good opportunity there to perhaps do a needs assessment or to do a study of how acceptable data like this would be to parents or to neonatal clinicians.
And to bring in the parent voice to be part of that education, that patient education.
Right.
And the clinical education too.
Right.
So, yeah, I see when I'm hearing all of this, my brain is going, okay, I see a work group, I see a paper, I see another paper, I see Wakako asked a question.
Please.
It's not really can you hear? It's not really a question, but that discussion brought me back something that, and I'm just being a devil's advocate, and hearing about oncology application of this external data in oncology. I myself is but I benefited highly from that type of, and that's probably the only reason why I'm still alive. But the treatment I received was not effective for everybody. It was a rare thing. So it brought me back the questions I asked. I knew if I enrolled in this, I'm going to get this 10 months long thing. And I remember the specific question I told the team, this is me. It's about me. It's so easy to make a decision. Immediately, I thought about the NICU parents that have to make decisions for their baby, that they have to live with the decision they make the rest of their lives, right? So that experience of 20 years ago always looks back at me. And but my important question was that at least did you test it enough in animals or labs or cells or something? And you believe, 90% of your heart believe that this is a better. So most parents think if they hear clinical trial, they have underlying assumption that what is offered is better. And they don't think about potential harm, like Ralph, I think it was you that mentioned that. So yes, acceptability, access, all the things that have been mentioned. We need to build a good consensus among us. A place like Inc. would be perfect because it could potentially cause a lot more confusion in vulnerable parents' mind. It's just my thought.
Yeah, and it's a great one. And it actually ties together a couple of things that Deb and Sheila mentioned. Solutions are not solutions if they're not adopted. And this is something that the team at C-Path hears me repeat ad nauseum. They're probably tired of me saying that. One avenue is the fact that we have the community. We have the different perspectives to make and transparently building those solutions for everyone to see, and maybe even with some formal regulatory review processes just for the solutions, not the medical products, but the solutions themselves. That's what guarantees that there will be some adoption, but it also provides the opportunity to open the door to show the tangible case study, the tangible something that is not abstract that you need to explain for 20 minutes and people go, I don't understand any of this. So yes.
I would like to elaborate a little bit more what you said, because Thomas Salatz, which is known for his score over here, also did some work on long-term safety, we called it that way, in parents. So how do parents feel 10 years after recruitment in randomized controlled trial, parents of neonates? So once they know the outcome, their child could have been died, could have been majorly impaired, or it could be okay. And actually parents felt well about it. They were proud and they were comfortable with their decision 10 years ago. So that was the good news. The bad news was for us. A lot of them didn't recall randomization. I agree that they felt it can only be better to be in a study. So let's say the genuine optimism that the study is better than doing nothing. So there was also quite some interesting information for us as a research unit, because we felt that we explained it quite well, but actually retention 10 years later wasn't that great. About 50% of the parents was no longer aware that indeed it was a randomized controlled trial. So people forget, and actually we all forget. If you would ask to you, where have you been 10 years ago? You may also have forgotten your summer holiday. So it's important information, but parents in long term feel comfortable with the recruitment also if it is randomized controlled trial. And some of these studies, like the insulin study, was in there, and the intervention arm was poorer than the control arm. So it occurs. It does also quite interesting information, but not so performant for us.
But that opens up the door for a continuous dialogue. And you may not necessarily capture every single parent for a continued advocation, but you capture the adopters, and with the adopters, you can do a lot.
And knowing that we have stakeholders involved, it makes it much easier adoption across everybody.
Yes.
Across the entire team, across the patient. And basically to say, hey, I was a parent just like you. Blah, blah, blah. It's very
right. So with your closing statement, Deb,
Yes.
I'm going to take the prerogative of adjourning the session now, and we will go on a break. Awesome. Thank you so much. Please give a hand to the panelists. I enjoyed the conversation. Thank you.